Any guesses when it cut out?
I don't know.
All right, sorry about that. We're just going to give everyone just a quick moment to get moved over to the to this new stream. Maybe if the control room can drop a link to the new stream in general, that would be helpful. Oh, looks like maybe someone else beat you to it. Let's see. Yep, part 2. Is that is today's? Okay, excellent. So thank you so much for that, sharing that link to that new stream. I'm just going to give everyone just a quick minute to hop on over there. And if one of the instructors listening in here could share the just share that over on the forum, let folks know that we had an internet hiccup and we've started a new stream, that would be really helpful. I'm just gonna let folks switch over here real quick. I think Evan, you have a live count, right? Okay, so we'll just give it a minute here. We need some elevator music here. All right. All right. Well, perfect. Well, thank you everyone for bearing with that first technical hiccup in what, the first 5 minutes of the workshop. As everyone else around the world has been experiencing, dealing with these kinds of changes with respect to the new global pandemic way of doing business, is just, it's how it goes. So we'll keep everyone posted, if things like that happen again, and we hope that everyone can be flexible. So with that, I think I had just rounded out, before the feed got lost, I just rounded out my list of things that I wanted to go over. Most important was the code of conduct. And then I would like to kick off the workshop proper. So I'd like to introduce our first speaker of the morning. We have Dr. Greg Capariso. Greg is an associate professor at Northern Arizona University. He is the director for the Center for Applied Microbiome Science at the Pathogen and Microbiome Institute. Greg is also the principal investigator for the QIIME 2 project. Greg is my boss. He's an all-around good guy and love working with him. And really excited to hear what he has to say about kicking off this workshop. So with that, I'm going to hand things over to Greg.
All right, thanks so much, Matt. And can I just confirm that from the control room that you can hear me and that you can see my slides? Okay, great. Well, welcome everybody. I'm extremely excited to kick off the first online QIIME 2 workshop. I'm going to start this session today with a quick intro and acknowledgement. So some of the instructors, I'll introduce our instruction team and talk about our funding sources. And then I want to spend a little bit of time just sort of briefly talking about microbes and microbiome science. And for a lot of folks, that'll be review, but I like to do that just to sort of get us all on the same page in terms of some of the terminology that you'll be hearing over the next few days. I'll then start to wrap this session up by introducing QIIME 2, and I'll talk about current features and some upcoming features, some new, very exciting things that we have coming down the pipeline. We have assembled an excellent team of instructors. So we have 21 instructors and TAs for this workshop. These are folks from North and South America and Europe. So we've got a good global community here. We would love to continue to expand this. And so if you're interested in getting involved in these workshops in the future, keep in touch and I'll tell you how to do that. We would love to expand our development team or our instruction team even further around the world. Before I move on, I just want to give a special acknowledgement to two of the folks on this list, Matthew Dillon and Evan Bollian. Matt and Evan are research software engineers in my lab at Northern Arizona University. And in addition to being like core QIIME 2 developers, they handled all of the logistics and the technology for this workshop. And so it's very safe to say that without Matt and Evan, this workshop would not be happening right now. And so a virtual round of applause, please, for Matt and Evan. I also just want to take a moment to acknowledge our funders. We have direct funding for QIIME 2, and then we also fund QIIME through some indirect sources. For example, some projects that we run might also feed into QIIME 2 development. Our hosts for this workshop are the Chan Zuckerberg Initiative, who is providing the funding for the workshop, and the CABANA Group, who has really helped us build the global audience that we have for this workshop. And so thanks to all of them. And let's start talking about microbes. What's a better place to start than where it all began with Anton von Leeuwenhoek, who in the 1670s developed some very powerful microscopes capable of 200x magnification. And von Leeuwenhoek is using his microscopes credited with the discovery of the microbial world. He pointed his microscopes at basically anything that he could come up with. And so he was the first one to observe different types of human cells, bacterial cells, yeast cells. And these photos that I have over on the right were taken with a replica of one of von Leeuwenhoek's microscopes. And so these give a good picture of what our first views of the microbial world looked like. Now, as the centuries went on, our technologies for studying microbes expanded. And so we moved from, in addition to microscopy, we began growing microbes in culture to study them, and that includes things like monoculture and also polymicrobial culture, like Winogradsky columns. And so for centuries, this was our toolkit for studying microbes. And in the past couple of decades, our toolkit has expanded to include culture-independent approaches to investigate microbial communities. Now, these primarily make use of the fact that all cellular life has a shared evolutionary history and that some genes are shared by all organisms. Now, in many of our more recent studies, we'll use the sequence of some of these shared genes as a genetic fingerprint for different organisms. And so DNA sequencing has expanded our toolkit. And the way that this typically works, and so like if we think about a marker gene survey where we're looking at a single gene and thinking about using that as a marker for different organisms, what's really happening here is at some time in the distant past, eons ago, there was some organism that encoded this gene in its genome. And so I'm just representing a small fragment of a DNA sequence here. And as time progressed, mutations randomly accumulated. And present day, we can observe the differences in this gene sequence, and we can associate those differences with different taxonomic groups. And so in other words, we can get a taxonomic and a phylogenetic signal from those gene sequences. The primary, or I shouldn't say the primary, the most common gene that we've used for this is the small subunit ribosomal RNA. We use this gene frequently because it's ubiquitous, so all cellular life encodes the small subunit ribosomal RNA gene in its genome, and it contains some regions that are identical across diverse organisms. And so there are some regions of that gene through those eons of evolutionary time that have not changed at all. But it also contains some regions that are variable across organisms. So some regions that have changed a lot. And so the regions that are identical are essential for us to be able to isolate that gene from a complex mixture of DNA. And the regions that are variable then allow us to use that gene to differentiate taxa that might be present in an environment. And so the small subunit ribosomal RNA, while it is the most common, is definitely not the only gene that's used with this approach. So folks might use genes like NIFH, or they might use non-genes, other regions of the genome, like the internal transcribed spacer region, which is a non-coding region of the genome that is often used for profiling communities of fungi. We'll talk about these different genes a bit over the course of the next few days. And so our toolkit expanded. We now use sequencing of ribosomal RNA and other marker genes. And this really led us to our understanding that the world that we live in is dominated by microorganisms. So in this phylogenetic tree here, this was created by Norm Pace, and it's a 3-domain tree of life. And so it's showing the bacteria, the archaea, and the eukarya. And what I've done here is I've highlighted the multicellular organisms in this tree. And so here's us over here. Here's some of our relatively close relatives like mushrooms and corn and seaweed. And everything else in this tree, all of the other life on Earth, is single cellular organisms. These technologies have also led us to understand that microbes live in complex and diverse communities everywhere on Earth. And so that might be in our bodies, that might be in our soils and on our plants and in our foods, and in other environmental scenarios, like, for example, this hot spring in Yellowstone National Park, or in marine water, fresh water. And so microbes are everywhere. And what really motivates me, for example, to build things like QIIME 2 and to work on this, is the incredible opportunities that microbes offer to improve our world. And so we're now using microbiomes to improve the efficacy of medical treatment. So this is some research that has gotten a lot of press for it lately, where the microbiome has been shown to impact how effective immunotherapy is in cancer treatments. Moving beyond human health, microbes can also help us to reduce our impact on the Earth. So when I think of microbes, I think of them as sort of the great innovators. They figure out how to make a living off of all sorts of things. And we can, using these new technologies, we can learn from these microbes, and they might be able to help us manage some of our own waste and pollution problems and build more sustainable systems. For example, understanding microbiomes might lead to more sustainable crops. We can learn by studying and optimizing agricultural microbial systems. We can retain more water on our farms. We can retain more nitrogen. We can reduce our dependence on fertilizers and other off-farm amendments. And ultimately, a lot of people these days are interested in sourdough bread making and fermentation, and understanding microbes is going to lead to new varieties of food. And so there's just all sorts of applications of these technologies. And that, like I said, is really what excites me and motivates me in this area. So if we think about the different technologies that I mentioned, I like to think of them as sort of illuminating different regions of the microbial world. And so in this figure, imagine that I'm representing the microbial world with this gray box that I have. And the different technologies that we're using, like culturing, for example, are going to shine a light on certain regions of that microbial world. And if we start thinking about sequencing, so for example, like 16S sequencing of the V6 region of bacterial genomes, maybe that shines a light on a bigger area, a different area of the microbial world. And say if we look at different primers using still 16S sequencing, we're going to observe different regions of this microbial world. There's a couple things that I am trying to represent here with this diagram that I want to point out. Number one is that we're not getting all of it. So we are seeing some of this microbial world, but we are still not observing everything. Our technologies are always going to be biased toward observing some organisms and missing other organisms. The other thing that I like to point out is that we have to be very careful when we're comparing microbial profiles that are generated using these different technologies. And so, for example, if you use V2 versus V4 16S sequencing, you're getting a slightly different picture of the microbial world. You have a different opportunity to, there are different organisms that you're going to have the opportunity to see with those different technologies. Now, another technology that is, another sequencing technology that is very interesting here and helpful in this respect is shotgun sequencing. Oh, excuse me for the notifications popping up on my screen. And so shotgun sequencing is going to illuminate more of the microbial world. And so the idea here is that it is not subject to the biases that these different primers are subject to. And so by sequencing all of the DNA that's in an environment, rather than isolating just a certain region of the genomes, we will observe more taxa, more of this microbial world. But we're still probably not going to see everything. So we're still going to be missing things, for example, that our DNA extraction is not going to work well for. And so like we're unable to get the DNA out of these cells. And we're also probably still going to be missing some low abundance organisms. And so all of these approaches will typically favor the things that are present in high abundance in favor of things that are present in low abundance. And so I'm talking here about different ways of looking at the microbial world. If we look at geographic information systems as an analogy here, I like to think of this because this is something that a lot of people are familiar with, for example, through Google Earth or Google Maps, where we might be trying to model the real world. And we might think of this as different layers that we can observe or not observe. And so, for example, if we are trying to put together, say, like a new water infrastructure for a city, for example, we might need all of these layers. We might need to know where our customers are and where the streets are and what the parcels of land are, elevation, land usage profiles. But on the other hand, if we're planning a hiking trip, for example, maybe what we really need to know is elevation profiles and where the roads are so that we know how to get to our hiking trail and we know what that trail is going to be like. And so depending on what we want to do, what we want to model, we need different layers from one of these systems. Now, I think the same way about studying microbial systems. And so the layers in a microbiome survey are going to be things like taxonomy. And so that would be things like what bacteria and archaea are present in an environment, what fungus is present in an environment, what phage and other viruses are present. And so these are different sort of taxonomic layers that we could think about including in a microbiome survey. If we want to know what's the functional potential of this environment, and so for example, does this microbiome seem to have the ability to fix atmospheric nitrogen into bioavailable nitrogen. We might not necessarily get that from a taxonomic profile, at least not from a 16S profile where we're not going to have species level resolution. But we might be able to get that by sequencing the functional genes in that environment too, and assembling a pathway from genes that we observe. If we're interested in functional activity, we might want to think about including information like a metatranscriptome or a metaproteome or a metabolome. And so again, these are sort of the different layers in a microbiome analysis. So I want to wrap this section up with a couple of quotes here. This is a quote that I like to share from Thomas Watson, who was famously quoted as saying, he was the chairman and CEO of IBM, and he was famously quoted as saying, I think there's a world market for maybe 5 computers. Now, I probably, just looking around me here, I probably have 5 computers sitting on my desk right now. And so he was, sort of famously wrong about that. But I don't want to be too hard on him because it's difficult to make predictions, especially about the future. And so, I think that we are sort of in a similar place right now with microbiome technologies. So we are just sort of scratching the surface of what we can apply these sequencing technologies to, for example. And I think we're going to see a big expansion of what we're using those for in the future. And so now what we think of these as being things that, we're using in labs, like in the building that I'm in today, we've got sequencing instruments just upstairs from me. In the future, maybe hobbyists will use these too, for looking at home gardening and optimizing soil microbial communities, or in home brewing, for example. And I'm going to go out on a limb and just make some predictions of my own about the future. And that's that we are really going to start to see the promises of microbiome science take hold when we start thinking about integrating all of these existing technologies and some of the newer technologies that are starting to make their way into microbiome surveys. And so, for example, using mass spectrometry to study the small molecules that are present in an environment. Highly multiplex bacterial genome sequencing to help us understand the full genome composition of some of these organisms. And it's not that one of these technologies is going to replace the other. It's that all of these technologies are going to be integrated to give us a bigger picture of the microbial world. All right, so let's change gears a little bit and let's talk about QIIME and QIIME 2. QIIME 2 is, like Matt mentioned in his intro, a project that I'm the PI on. And so my lab in Northern Arizona leads the development of this, but it's really a community effort. And that is well illustrated by, for example, the author list on the QIIME 2 paper that we published just about a little over a year ago now. And so we have on this author list about over 100 co-authors from over 70 different institutions in 10 different countries. And so while I see us as taking on the sort of leadership responsibility for this community, this is really a large community effort. And there's space in here for new leaders. And so we would love to get more people involved, more people teaching these workshops and leading the development of different components of the ecosystem. And I'm going to tell you how you can get more involved with QIIME 2. At present, there's over 23,000 citations for QIIME or QIIME 2 in the primary literature. And for me, it's just incredibly exciting to see all of the things that people have applied this to, which were never things that I had imagined when we first got started with this project. So QIIME started as a sort of postdoc project for me when I was in Rob Knight's lab, which was then at the University of Colorado. And we were developing this to apply to some of our own surveys. But we also figured that we wanted to make this accessible to other users or other researchers around the world. And so, again, we're just incredibly excited about all of the cool things that people have thought to apply this to that we had never thought of when we were just getting started. Chime and Chime 2 is completely free and open source, so you can use this for any purpose. You have a licensed right to use, redistribute, and even modify Chime 2 if you want. What we would hope is that if you're interested in this and you're interested in building software and making modifications, that you would get in touch, and you would do that by joining the Chime 2 community and contributing. We, like I said, have a worldwide network of collaborators. Now, I want to mention this quickly. First, I just want to thank everybody who filled out our pre-workshop survey. We noticed, looking at the initial survey responses last night, that about 25% of the folks mentioned that they occasionally use Chime 1. Nobody said they use it daily anymore, but some people are occasionally using it. And so it's just important to mention that Chime 2 has succeeded Chime 1. As of January of 2018, we don't support Chime 1 anymore. I have a blog post linked from this slide where we talk about exactly what that means. And so if you need to, for example, reproduce something that somebody else did or that you did with Chime 1, you can still download those virtual machines, you can still run it. We just don't support it anymore. Chime 2 is much better in every way than Chime 1. So if you haven't switched yet, I hope that you're sort of viewing this workshop as your opportunity to switch over. You can install and use Chime 2 on your own computer or on cloud resources. You can install this natively on your machine, whether it's Mac OS, Linux, or Windows. As long as you have Windows, your machine is capable of Windows subsystem for Linux. And we have virtual machines that you can use locally if for some reason you're not able to install Chime 2 on your machine. And so I'm going to talk now about some of the current features of Chime 2, and in particular, some of the things that attract users to the system. I won't spend a lot of time enumerating methods and visualizations that you can access through Chime 2, because you're going to spend a lot of the next few days looking at all that and working through some of those methods on your own. So I'll talk a little bit about how we make Chime accessible and about the community that we have built about it. Really briefly, though, a big focus of Chime 2 and what draws a lot of people to the system is that we focus on making the latest methods available and interactive visualizations that will help you explore your systems. These are just a few examples of some of the interactive visualizations available in Chime 2. One of the main things that attracts people to Chime 2 is the community that we are building around it. And at the core of this community is the Chime 2 forum and our community code of conduct. Matt mentioned our code of conduct briefly. We adopt that code of conduct in all of our community interactions, including this workshop. So it's definitely something that you need to read when you're just getting started. You should also go ahead and create a free forum account. This will be your main place following the workshop to get technical support with Chime 2. We do try to get back to all technical support questions within one working day. And there are other types of discussions that you can have on the forum as well. And so we have a job board on here. I'll mention this again in just a few minutes. We also have general discussion, and so if you want to talk about primers and things like that, you can feel free to post those types of questions to the Chime 2 forum. The forum is also a place where community members post resources that they have created. A really nice example of this, and one of our most popular posts on the forum, is this translation of some of the Chime 2 documentation into Chinese. This was contributed by a community member, and this was done actually before we published the Chime 2 paper, and so we invited this community member to co-author the paper with us. And that's really like one of our approaches for trying to build this community. So if you, when you join the community, you're part of this team, and we will try and involve you as much as we can. Our workshops are also a great illustration of our community. And due to the global pandemic, we are now transitioning our workshops online. And so like Matt mentioned, this is our first online workshop. This is something we've been wanting to do for a long time, but the workshop has sort of, the pandemic has sort of pushed us to really make some progress in that direction. In our in-person workshops, we would have hands-on sessions. We would have sessions where users can interact with each other. We would have small working group discussions around topics of interest for smaller groups. And our goal here over the next few days is to try and recreate all of those experiences in this online setting. And so you'll see how we're going to do that. The hands-on stuff, we will be working you through working through a tutorial with you that you'll be doing on your own computer. We are working on grouping students into what we're calling pods, but like smaller groups where you can get help or get your questions answered and hopefully interact with one another and make some new connections there. And we're also going to try and facilitate some smaller group discussions through what we call water cooler chats. And that's sort of an idiom that we use in the US to sort of mean the informal conversations that you have when you go get a glass of water while you're at work or a cup of coffee while you're at work. And so we're going to try and recreate that here on Slack. And so if a topic of interest to some students comes up during a session, we will try and move that over to Slack during one of the breaks where we can have either video or just a text-based chat about that topic. And so by coming to this QIIME 2 workshop, you are joining the QIIME 2 community. There's a few other features that are less immediately obvious in QIIME 2, but that are very exciting and really enable some of our core goals for this system. And so I'm going to talk about those right now. The first one is our provenance tracking system, which many of our users discover after they've started using QIIME 2, and which they're extremely excited about then. QIIME 2 is also available through multiple user interface types, and it's built on a plugin architecture. I'm going to talk about what all of those mean right now. So our retrospective data provenance tracking system is really designed to help you answer the question, what did you do 5 months ago? And so these are actually some notes that I have from a QIIME 1 analysis that I ran many years ago. And this is, maybe what a typical folder looks like on my computer where I'm running a QIIME analysis. And so in this case, I was doing pretty good. I was keeping notes here about what commands I had run. I had all my files in this directory. But sometimes or almost all the time, you'll do an analysis, and then months later, you'll need to come back and figure out what you did. That might be because you've submitted a paper for publication and a reviewer is asking you some very specific piece of information. For example, what version of NumPy was installed on the machine where you ran this analysis, because there was a bug that may have impacted your results. Or maybe you are now doing another analysis and you want to come back and apply what you did in this previous analysis in your new analysis. So now maybe you can poke through all of this and you can figure out what you did, but maybe you don't have some key information here. Like maybe you forgot to record what version of NumPy you were using, or didn't even know that that was something that you were supposed to record. Or maybe you come back and you can't figure out, was I using sample metadata or sample metadata.1? Or what is this one with the fixed missing controls? Which of these is the version that I was using? Well, QIIME 2 tries to help you address all of those issues by recording all of that information for you behind the scenes. And that's what data provenance tracking is in QIIME 2, and it ensures that your analysis is going to be reproducible, either for you or for somebody else. And so we will work you through some examples of looking at the QIIME 2 data provenance, but really briefly, anytime you have created a QIIME 2 result, so you've created something with QIIME 2, whether that's a visualization or it's some intermediate result like a phylogenetic tree or a feature table, you can view the data provenance for that artifact. And so this is, or sorry, for that result. And so the data provenance is going to tell you all of the steps that you took to generate that. And so in this version here, this circle down here would represent this visualization that we're looking at, and everything above it are the steps that got us to that visualization. And so this box here is showing that this was an action called denoise single that was run through the Data2 plugin. We can see what parameters were used when we ran this. And this also includes a ton of additional information. And so it actually would include information about what other software is installed in this environment. For example, including NumPy, which might be useful if you're trying to answer that reviewer's question that I suggested. Okay, so QIIME 2 also is available through different interface types. And these different interface types are geared toward users with different levels of computational sophistication. And so right now we have a prototype graphical interface. We also have another graphical interface that's in the works that I'll mention shortly. But these are really geared toward folks who are sort of on the domain scientist end of the spectrum. And so maybe you don't want to use a programming language or command line, but you want to be able to run the same analyses. We're going to focus in this workshop on the QIIME 2 command line interface. And that is a very powerful interface that allows you to easily run this on cluster computers, for example, or on your own computer. And so we will be spending our time in the workshop talking about this. On the other end of this spectrum here would be sort of like the data scientist interface. And that is a Python 3 API that you can use if you are trying to automate QIIME 2 workflows, either, for example, in your own Python code or in interfaces like a Jupyter notebook. Now, there's one more interface that I have up here that I didn't mention yet, and that's QIIME 2 View. This is another one that you're going to get some experience with during the workshop this week. So QIIME 2 View is a read-only web interface for viewing results without having QIIME 2 installed. And so this is how we're going to allow you to interact with QIIME 2 results while you don't have it installed on your computer. This is also really helpful if you want to share results with others. And so, for example, if your boss or your PI doesn't have QIIME 2 installed and they don't know how to use it, but they want to be able to interact with some of the results, you can share those results with them using QIIME 2 View. The last thing I want to mention in this section is the plugin architecture of QIIME 2. So all of the bioinformatics functionality in QIIME 2, everything that we use for studying microbiomes, is implemented in a QIIME 2 plugin. These are written in Python 3, but the underlying methods can be written in anything. They can be written in R, they can be written in C or C++, and then Python just becomes this sort of annotation. Sorry, I'm getting some more notifications popping up here, but I'm going to do my best to ignore those. And so so this is really like how we plan to keep pace with the field. And so we don't need to be an expert in all of the methods, and we don't need to be developing all of the analysis functionality in order to make that accessible to QIIME 2 users. And so, for example, the Data2 plugin, which I have an example of up here, is a quality control tool that we will get some experience with in this workshop. This is developed by one of our collaborators. You can use Data2 independently of QIIME 2, but you can also use this within QIIME 2 because there's this Python plugin, or this QIIME 2 plugin for it. Now, I noticed reviewing the workshop responses that about 50% of the respondents indicated that they use a programming language like R or Python 3. And so I would definitely encourage you to to start looking into these Python plugins or QIIME 2 plugins and consider building QIIME 2 plugins for some of the methods that you're creating. The QIIME 2 library is the primary site where you can where you can learn about what new plugins are becoming available. And for potential plugin developers or existing plugin developers, we have lots of new functionality coming online there. And that will help you, for example, do things like automated testing of your plugin or package building. The QIIME 2 library is a work in progress right now. It exists and you can find a lot of very cool plugins there, but there's just tons of cool new stuff coming soon. And that work is funded primarily by the Chan Zuckerberg Initiative. Okay, so that's a good segue into what's next for QIIME 2. And so where we're really going with this, I talked before about the microbiome layers. QIIME 2 is going to become a microbiome multi-omics platform, and that is going to be enabled by plugins. So we are going to, and we currently are working with people who are developing metabolomics plugins and shotgun metagenomics plugins. And all of these will ultimately be accessible to you through the interfaces that you're most comfortable using with QIIME 2. Another very exciting feature that is on the horizon is provenance replay. The idea here is that you will be able to generate, so we looked at this data provenance and I described how that fully documents the workflow that you ran. Well, what we're going to do is we are going to enable the programmatic extraction of that information so that you can generate an executable code from the data provenance. And so that will allow you to rerun analyses that you ran before in a single step. And so in this example, maybe you're taking this workflow that you previously used to create taxa bar plots and then automatically generating your pipeline as an executable file that you can run on some new data or rerun on your old data to make it quicker to redo an analysis. There's also going to be many new ways to use QIIME 2. That includes through Galaxy and the common workflow language. So Galaxy, we will have all QIIME 2 functionality and plugins accessible through a Galaxy interface. And so if you're interested in graphical interfaces, this is really where we're focusing most of our effort right now. So that is on the horizon. We're actually getting pretty close to having an initial release of that. Common workflow language, we have an interface there that's ready for testing. And all the folks out there who are R programmers will be excited to know that we have a full R API that is now funded, and we're in the early planning stages of that development. This will allow you to run any analyses, any QIIME 2 analyses through R. And so the real key with all these different interfaces is that regardless of which of these interfaces you're most comfortable with, you can access all of the QIIME 2 functionality. If you are an R programmer, in the meantime, you can try using QIIME 2R, which is a tool that is developed to allow you to load QIIME 2 data in R and then analyze it with other tools. So for example, like Phyloseq. We have lots more on the horizon. I posted something on the QIIME 2 forum recently about this. If you want to read the full post and get some more details, there's a link down at the bottom of this slide. And so I just want to wrap this up by letting you know that in order to enable all of this new functionality that I'm talking about, we are currently hiring software engineers, postdoctoral scholars, and graduate students to join the QIIME 2 team. So if you're excited about QIIME 2, if you're excited about microbiome research and open science, get in touch with us. You can find more about this when we make the formal announcements. We'll do that on the QIIME 2 forum job board and on Twitter. And so keep an eye on those. Go create your forum account. And I will wrap up there, and we can maybe take a couple questions from this initial session if we have time. Thank you very much.
Thank you, Greg. So don't have any questions in just yet, so I'll let folks kind of sit and think about that for a minute. And while they do, and I should just mention, if you have questions, please share them on the general channel on our Slack. And so while we let that sit and wait for just a minute, I have a few announcements that I'd like to share. So unfortunately, Slack is currently experiencing system-wide outages. I shared a link in the general channel on Slack. Maybe people have received it, maybe not. Unfortunately, that is outside of our control. We will try and keep you posted as they come back online. But many of their services are down or degraded right now. So we'll just kind of have to work with that as we go today. As well, we were able to get our hands on a new and improved internet connection for our control room. So we will be switching over to that at the break. But if we run into any more like internet speed technical difficulties, we'll switch over sooner. I also just wanted to mention that there is a slight delay on the video feed as well. So if it seems like we're taking a bit longer than anticipated to get back to you with answering these questions, that's why. There's a slight delay to the feed. And then the last thing that I just want to mention here is we will have our first break of the day in about 60 minutes, 60 or 65 minutes here. So if you're starting to wonder when that first break is, it's in just about an hour. And so with that, I'm going to just jump over real. Pardon? Oh no, you're right. Sorry about that. It is in 30 minutes, not an hour. We will be back in an hour from break. Yeah, so the first break will be in about 35 minutes. Thanks for the correction there. So let's see what we have over here in Slack. There's a couple of questions coming in here that we might not answer here in this live Q&A session. They might be better reserved for later at the end of the day when we hold general questions and answers. So if you have questions specifically about what Greg just presented, that would be good timing. Just wait a moment here.
Matt, I do see one. I'll go ahead and answer that. So I see a question that says that the integration of different omics approaches sounds exciting. Any timeline for this? This is in progress right now. And so if you were to go to library.chime2.org, you would see like we have a metabolomics plugin. We are working on some shotgun functionality. And so this is really, the way that we do something like this is we're sort of working on it right now. And these things will become available. We'll be announcing them on the Chime2 forum. And so, I don't really have a specific timeline for when we'll be done with all that work, but that's probably because, like most things in research, we're never done with it. It's always going to be sort of evolving. But yeah, there are different things that you can do now beyond marker gene surveys.
All right. Maybe we will move on to our next section of the workshop here. Does that sound okay? Okay. So we will we'll start shifting gears here real quick. So the next section that we have here is titled Semantic Types and Data Formats. And this is a pre-recorded video that we're going to share with you. And the author of that pre-recorded video is yours truly. So at the end of that video, we'll have some time to take questions and answers before we head off to our break. So with that, control room, I think we're ready to get started there. Hello, my name is Matthew Dillon, and I'm a senior research software engineer in the CAPANA lab. I'm one of the core QIIME 2 developers. Greg just gave us a great overview of a lot of the more interesting bits of functionality for QIIME 2, and we're going to get a chance over the next several days to really dive in and get a closer look at how a lot of this works by conducting a hands-on microbiome analysis using a tutorial data set. There are a few core concepts that we think are important to start thinking about now. And these have to do with kind of the language of QIIME 2 and the way that we think about data in QIIME 2. So I'll start with an example. It's a little bit of a contrived example, but maybe everyone can relate. So imagine, sit down with your afternoon cup of coffee or tea and open up your email and just try and catch up on things, and you get an email like this. It says, urgent analysis needed by Tuesday, 2 p.m. And it looks like it's from my colleague, Evan Bolian. And it says, hi, Matt. Hope everything is going well with you. I was just wondering if you could analyze this data attached really quick. I need it by Tuesday at 2 p.m. There's a few files in here, but I just need you to check into the sample 11E by total one. So I look and I see there's a data.zip attachment, and I unzip it and I open up sample 11E by total, and I see something that looks like this matrix of numbers. There appears to be a couple of things that maybe look like labels here, A, B, and C, and S1, S2, and S3, but there's no description anywhere in this email about what Evan has provided to me. Not only that, but there's also no explanation really here of what Evan needs. All we know is that he wants us to analyze this data, and he needs it by Tuesday at 2 p.m. So I think it's safe to say we are missing important context here. We don't really know what he wants us to do, and we don't really know what the nature of this data, this matrix or this table represents. So I've got a couple of potential scenarios here. These are different options that we could take to move forward. And I want us to think about some of the impacts of that missing context in Evan's email. So the first scenario is I could just take that table, that matrix, and try and do something with it. Attempt to run some kind of analysis. Maybe I work in a microbiome lab and I know that there are certain kinds of analyses that we tend to do, for example. But because I don't know what Evan has done to generate that data, and I don't know what that data represents, I might do the wrong thing. And so the outcome, or the potential negative outcome here, is that I derive incorrect conclusions. So I might run some analysis that I think is right, turns out has some fundamental flaw, and then I send the results back to Evan and we publish the wrong data, or we make a potentially costly incorrect conclusion out of it. Another scenario here. I could look at Evan's email and say, well, this guy, he's asking way too much and has not given me any information. So I'm going to go back to him and say, hey, Evan, I need more details. I need that missing context. The risk here is that we missed that deadline. He made it very clear that we had a limited amount of time to do this work. And if I have to have a back and forth discussion with Evan several times, kind of checking to make sure that I understand something or asking for clarification, we could miss that deadline. Another scenario that we could run into is a little bit more subtle, but we could do what we usually do with these data. So imagine that I have been working with this group for years, and we have a very kind of well-established pipeline for running whatever kinds of data it is that we run. And so I just do my normal thing. The risk here is that we might not necessarily be using the latest techniques available to us because we don't necessarily know all of the details about these data. Okay, so a common aspect to all of this is that we're kind of talking about context in terms of this email conversation. But now I want us to shift our focus and think about context with respect to a computer. I have a minor complaint here about how we often think about and talk about computers in the sciences. I think oftentimes we imbue this almost godlike power in computers, that somehow a computer is all-knowing or is capable of understanding what it is that you want to do without you even telling the computer. So I've got this still of some kind of oracle-like computer type creature here from one of the Matrix movies, and this bothers me because I think computers are a really great tool, but I think they're only as good as we are, the people that program it or the people that are using it. So another way to put this is computers are not clever. They will only do exactly as they are instructed. They can't read between the lines. They can't infer things about what it is that you want to do or what it is that you're after. So with all of this in mind, I want to pose this question. What kinds of opportunities are created when we're able to provide more context to a computer? And let's kind of think about this first in terms of this email example. So if we revisit these scenarios that we were looking at a few moments ago, that first scenario where we potentially do the wrong thing and generate incorrect conclusions, well, if we had more context from Evan there, then we could have been prevented from providing the wrong input to the command. That would have been really great if we were able to somehow know, okay, this data does not satisfy some critical assumption of this analysis that I was about to run. For the second scenario, the one where potentially missed the deadline, the advantage of that additional context there is that we would not have to go somewhere else to find the context. The contextual data is somehow already encoded within that message or within that email. So the idea here is we have a complete file of everything, so to speak. And in the third scenario, with additional context available to us, this one's a little bit more of a hypothetical, but this is a long-term goal in our eyes. But being able to ask a search engine, not just like what should I do with this data, or run some type of analysis, but to be able to actually ask like, what can I do with this type of data, sounds really, really interesting to me. Being able to search in some big global search engine or database and learn about all the different types of analyses that I could conduct with some type of data, that sounds pretty exciting to me. Okay, so with all of that set up, I imagine you're all kind of anticipating now that I'm going to show you how this relates to QIIME 2, and that is exactly the case. So QIIME 2 attempts to encode useful context within some related concepts in the framework and within the ecosystem. So we're going to look at these two things one after the other. So again, the title of my talk here was about semantic types and formats. So let's start with semantic types. So what this means here, the term semantic types, it's the vocabulary for describing what data, quote, is So what I mean by that is the semantics of the data refer to the meaning or the concept of the data. So when we talk about, for example, like a phylogenetic tree, we're not necessarily talking about some tree that is sitting somewhere, maybe in someone's garden, with labels about the various species or whatnot on this tree. We're talking about the concept of a phylogenetic tree. It's not something that you can hold in your hand, but it is a concrete conceptual idea. And so with these semantic types, the idea here is that it's a language in which we can talk about different types of microbiome bioinformatics data. So I've got a few boxes here on my screen, and these all represent different types or common types of semantic types represented within a QIIME 2 environment. So these are things like a feature table of frequency, or a phylogeny, or a sample data of sequences with quality, or a feature data of taxonomy. Now, if none of those words mean anything to you, if you don't know what taxonomy is, or if you've never heard of a feature table, that's fine. That's the point of this workshop. We're going to start to introduce these concepts over the next few days, and we'll revisit this. But maybe some of you in the audience are familiar with these concepts, like you've received, say, sequencing product from your sequencing center. You might recognize then that this semantic type here, sample data of sequences with quality, well, that describes that data that you've received from your sequencing center. So here's the other side to the coin, data formats. So semantic types refer to the meaning. Data formats refer to the way that data is represented. So what it looks like in a file, or what it looks like in a computer program, how it is actually laid out. So the important thing to keep in mind here is that semantic types keep the formatting separate from the meaning of the data. So we treat these as two separate things. So I've overlaid a few boxes here over my list of semantic types to highlight a few possible formats that could be used to represent data of that semantic type. So for the first one here, this yellow one, it was a feature table of frequency. We could represent these data as a biome file, or perhaps a TSV, that stands for tab separated values. That's like a spreadsheet. Or a CSV, it's a comma separated values file, similar to a TSV. Or perhaps it could be a pandas data frame, or an R data frame, or something. As well, here for the sample data of sequences with quality, we have the possibility of representing this data as a FASTQ file, or maybe as a FASTA plus qual file. There is no reason why one is necessarily better than the other, but the point is that different types of data can be, or one type of data more specifically, can be represented with multiple different formats or representations. There's also an interesting observation to highlight here as well. So you might notice that this feature table frequency and the feature table taxonomy down at the bottom both have many of the same formats listed here. So biome, TSV, CSV, or data frame. And then down here over the feature data of taxonomy, we have TSV, CSV, pandas data frame. You notice many of those in those two lists overlap. They're the same. And so I think the important thing to realize when you see that is this means that if I give you a TSV file, you can't just immediately look at that and know, well, yes, that represents taxonomy data, or that represents a feature table. The point is, the semantic type is that context that helps identify what the contents of that file actually are. So another really nice advantage to separating out the semantic meaning from the data representation is that formats can change over time while the semantics remain the same. So I have a figure over here on the right of several tools. Semantic type of these tools, if I was to come up with a name for this, is I would call these hand tools for woodcutting. And so that's listed down here in the bottom left in this blue box. And the intent or the meaning of the semantic type is that these represent a class of tools that are specifically used for cutting wood. And you'll notice this has nothing to do with programming, nothing to do with microbiome bioinformatics. Now in the figure, you see a sequence of 5 different tools. Now, all of these tools represent a different format or a different representation of a woodcutting hand tool, but they are all different. So there's this nice kind of evolution here from a primitive stone axe to a bronze axe to a modern axe to a crosscut saw, and then finally over here on the right is a gas-powered chainsaw. The idea that formats can change over time is, I think it's an important one. We learn more about how to represent data or how best we can, say, shrink file size or increase the utility of a different format. And so this is a really nice side effect of separating out the semantic meaning, hand tool for woodcutting, from the format, these various ways in which to do it. This also allows us to guarantee backwards compatibility. So results generated today in QIIME 2 will be backwards compatible with future versions of QIIME 2, or more specifically, those future versions will be backwards compatible with results generated today. So the last point that I want to emphasize here is that there is no one right way to do a QIIME 2 analysis. This is something that comes up every now and then. People want to kind of like abstractly talk about a QIIME 2 pipeline. And this is maybe not the mindset that will get you the most utility out of using QIIME 2. What I mean by that is QIIME 2 is the model for QIIME 2 and for the architecture of the whole thing is to provide a series of small modular functions or commands that can be composed together in some sequence or some order that is most relevant or appropriate for you and your analysis. So how commands work in QIIME 2 at a very high level is like this. The black box in the middle here represents some kind of QIIME 2 command. We'll call this a QIIME 2 action. And we talk about the inputs or the things that need to be provided to this action by using semantic types. And then the things that are returned from that action, we talk about those by using QIIME 2 semantic types. So we say this action takes semantic types of a certain type and returns outputs of some semantic type. The idea then is by using this language of the semantic types, we can start to glue more and more of these actions together. Now, another thing that you might be wondering about with all this discussion of semantic types and formats is how does the data actually work in QIIME 2? Where does my data go? What does QIIME 2 do with it? And this is where something pretty exciting comes into play here. So we have 2 different file types that we work with, and we'll talk a lot more about these in the coming days. But basically, QIIME 2 is able to generate two kind of high-level outputs, either artifacts or visualizations. Artifacts refer to data, and visualizations refer to something that we should be able to kind of look at or interpret. The kind kind of interesting idea here, and we'll get some examples of it as we get into the tutorial, is that artifacts are used as inputs and outputs for various QIIME2 actions, while visualizations represent a terminal output. So a command that generates a visualization is kind of the end of a branch of some part of your analysis. So a question that often comes up then is people want to know, well, where did my FASTA file go? Or where did my biome file go? When they see their QIIME zipped artifact. And the kind of exciting thing here for me is that it's just inside a zip file. We aren't inventing any kind of new formats. All we do is we zip all the files up as QIIME2 saves them. And so then this allows us to do a lot of pretty interesting things. If you're curious about learning more about the sort of specification design of this, there's a link here to our developer docs that you can look at. But kind of the most interesting thing in terms of long-term archival storage is because these are just zip files, even if QIIME2 goes away and doesn't exist anymore, these files can be opened with any modern zip implementation. You might have to, for example, change the extension from .qza to .qzv, but they are accessible that way. So the next question that we usually get is like, well, why zip the files? Why not just save the FASTA or save the biome file? The reason is because QIIME2 saves all of that context that we were talking about at the start of my talk. And that's what is represented here in this figure. The point is there is a lot going on here in order for QIIME2 to be able to keep track of things like decentralized provenance or information about what version of QIIME2 generated this particular output. So all of these boxes in here represent either a directory in the zip file or a file within the zip file. And most of this has to do with that context that we were talking about earlier. Only this tiny little section down at the bottom with these yellow boxes actually represent the data. So this is why we have QZAs and QZVs. And we will review this many more times over the coming days, and we will also answer any questions as you kind of let this sit and you think about this. Feel free to ask questions at any point, and we will gladly discuss. With that, thank you very much, and I will see you later. All right.
Okay, welcome back, everybody. And thanks for that first video lecture from Matthew Dillon. We have a couple of questions, or one question rolling in. Matt, user asks, if I have an HTML file, can that be entered into QIIME2 to take advantage of provenance? And the specific example this user is looking at is they have a Krona analysis of 16S data.
Yeah, that's a great question. The short answer is not without a little bit of work. So this would be a really good candidate for like a QIIME2 plugin that had a QIIME2 visualization. So QIIME2 plugins are the way that you could share different bits of functionality. So things like a visualization or a particular command or analysis. And the QIIME2 framework, which Greg talked about a little bit in his first section of the workshop here, the framework is really what handles all those things like the decentralized provenance tracking and allows us to generate the different user interfaces. But unfortunately, with just an HTML file, you're not able to use that in like QIIME2's provenance tracking. Any additions to that from the control room?
I guess Yeah, the one thing I would add is like that's kind of one of the real core things that Chime 2 offers developers. And so if, for example, like the Corona developers wanted to make that accessible to users, that would be something that the Chime 2 framework provides them. I don't see any other questions popping up in here for this session. So maybe we should move on to some of the announcements you had, Matt.
Yeah, that sounds great. We're going to break now. We have our first morning break, or at least morning for the kind of core Chime 2 team based in Arizona. So before we do that, though, now would be a really good time to make sure that you have installed Google Chrome and have installed the Chrome Secure Shell app. We shared some links to that in the slide deck today. Also, I sent that along in a few emails. But if you're running into any issues with that or need some help, hop on over into General and just let us know. We are also going to need those PDF sheets for the next session after the break. So I sent out PDF sheets to everyone. Greg, can you show us what those look like?
Yep, so I'm holding an example that I printed out of one of these PDFs, and so you should have a PDF that was sent to you on Slack that looks like this, and you'll see some specific information in these cells up here. So mine are blank because this is our template, but you'll find a username in there that we've assigned to you and various other information.
Great, thank you, Greg. So if you had any issues opening that file or didn't receive it or received the wrong one, just let us know in the general channel. So with that, well, let's just start our first break a few minutes early here. Take a few minutes, stretch, look away from your computer screen, get up and get the blood pumping, and we will be back in 35 minutes. Yes, so thank you. Yes, so we're going to switch over to a more robust internet connection. We're hoping that the YouTube stream doesn't drop, but if it does, we will share a new YouTube link in the general channel. So, yeah, thank you. Hello, welcome back. Hope everyone got a chance to get up and stretch their legs and maybe look at something besides a computer screen. We are going to jump into our first section of the hands-on components of this QIIME 2 workshop. And so I've mentioned a few times now that we sent out some PDFs with login information. Now is the time that we will need that. So make sure that you have that file pulled up. Maybe download it to your computer just in case, since Slack has been giving us some issues today. And what we're going to do is we're going to show you a very short 4 or 5 minute video from one of the QIIME 2, the core QIIME 2 developers on our team, Andrew Sanchez. And he's going to walk us through step by step how to connect to the server and sort of just orient us with Google Chrome and the Chrome Secure Shell and so on. Then what we're going to do is at the end of that video, we will pause the live stream here, and all of the instructors will be available to come in and provide hands-on support in the individual pods. So before each person's name in the Slack channel, you'll notice something like pod 02 or pod 05. That is the pod that you are in. And so we would potentially do small like screen shares or video chats in those pods to just kind of help make sure that everyone gets the support to move on here. So we've budgeted about 45 minutes for this. We expect this is kind of going to be the most complicated step of getting set up, mostly just because it's potentially new technology, new tools, and so on. And so if we need to show the video again multiple times, we can do that. It's, like I said, it's very short. It's 4 or 5 minutes. And with that, I think we will go ahead and pull this up. So feel free to follow along with the video while it's playing. And then if you run into issues, we'll chat with you in the general channel as soon as the video's done. Okay, thank you.
Hi everyone, my name is Andrew, and I'm going to teach you how to connect to the Workshop cluster server using the Chrome Secure Shell app. I'll be teaching you a few other tutorials as we go, so I'll introduce myself now. My name is Andrew Sanchez. I'm a research software engineer at the Pathogen Microbiome Institute, and I'm a new member of the CABANA lab and a QIIME 2 developer. We're going to learn about the Secure Shell app, what is it, why we're using it, how to access it from your Chrome web browser, and how to log into the Workshop server. So the Secure Shell app is a Google Chrome app, and we're using it so that you don't have to worry about installing QIIME 2 on your own computer or on another server. All you have to do is log in to our Workshop servers using the Secure Shell app, and you'll have an environment that's ready to go with QIIME 2 already installed, so that you can get up and running and start learning right away. The prerequisite for not only this tutorial, but the rest of the tutorials is that you've completed the required software instructions. You go to workshops.qiime2.org and you click the link for this workshop. Scroll down, you'll see the required software section here. Again, you should have already completed this step, and we're going to proceed assuming that you have. Let's get started. So, steps to connect to the Workshop server. First, we're going to navigate to Chrome://apps. Chrome://apps. Here you're going to see any apps that you have installed in your browser, and you should see the Secure Shell app here. So, click on that. Next, you need to fill in your username and hostname. Hostname is workshop-server.qiime2.org. Make sure new connection is selected here. Go down here to username. Type in your username and the hostname. You'll notice it's pre-filling in text up here. You don't have to do anything up here. After you type in the workshop server name, all you have to do is press enter. Please note that all this text has nothing to do with your computer. This is actually related to only the workshop server and this Chrome Secure Shell app. Here you'll see a prompt for the password. As you type it in, you will not see any action here. That's for security purposes. It doesn't want to show your password. So don't worry if you don't see anything happening there. Just type in your password and press enter. Again, all of this text here is coming from our workshop servers. So it says system restart required. There's nothing to do about that. Just once you see something like this, you're good to go. This will be different for you depending on what your username is. And congratulations, you've logged in to the workshop cluster server.
All right. We are going to take a quick break to jump in and help folks out individually as they're trying to log in here. So one thing that I just want to point out, it looks like there's a few messages that came in related to the password being incorrect. So one thing that you'll want to make sure to do is carefully select the entire password and don't grab anything else in your selection while you're doing that. It might be possible to grab some text at, say, the beginning or the end of that. So one suggestion that I have is maybe before you paste your password into the Google Chrome Secure Shell prompt, maybe paste it into a text editor just to make sure that you've copied the text that looks appropriate. And worst case, you can just type the password in manually, but that might be a little bit slower. And then also really good reminder in the general chat right now about remembering to trust the connection the first time you log in. So I don't think the video went into detail about that, but the very first time you log in, the server will prompt you with a yes or no question and ask, do you trust this server? And so we want to type out the word yes, Y-E-S, and then press the enter key. So actually, you know what we should do? We're going to play that video again one more time. Sorry, Evan, I know that wasn't quite what we discussed, but how about we play that video one more time, and then what we'll do is we'll send out a survey on general in the meantime to ask people if they've been able to successfully log in. So with that, let's watch that video again.
Hi everyone, my name is Andrew and I'm going to teach you how to connect to the workshop cluster server using the Chrome Secure Shell app. I'll be teaching you a few other tutorials as we go, so I'll introduce myself now. My name is Andrew Sanchez. I'm a research software engineer at the Pathogen Microbiome Institute, and I'm a new member of the Capareso lab and a QIIME 2 developer. We're going to learn about the Secure Shell app, what is it, why we're using it, how to access it from your Chrome web browser, and how to log into the workshop server. So the Secure Shell app is a Google Chrome app, and we're using it so that you don't have to worry about installing QIIME 2 on your own computer or on another server. All you have to do is log in to our workshop servers using the Secure Shell app, and you'll have an environment that's ready to go with QIIME 2 already installed, so that you can get up and running and start learning right away. The prerequisite for not only this tutorial, but the rest of the tutorials is that you've completed the required software instructions. You go to workshops.qiime2.org and you click the link for this workshop. Scroll down, you'll see the required software section here. Again, you should have already completed this step, and we're going to proceed assuming that you have. Let's get started. So, steps to connect to the workshop server. First, we're going to navigate to Chrome://apps. Chrome://apps. Here you're going to see any apps that you have installed in your browser, and you should see the Secure Shell app here. So, click on that. Next, you need to fill in your username and hostname. Hostname is workshop-server.chime2.org. Make sure new connection is selected here. Go down here to username. Type in your username and the hostname. You'll notice it's pre-filling in text up here. You don't have to do anything up here. After you type in the workshop server name, all you have to do is press enter. Please note that all this text has nothing to do with your computer. This is actually related to only the workshop server and this Chrome Secure Shell app. Here you'll see a prompt for the password. As you type it in, you will not see any action here. That's for security purposes. It doesn't want to show your password. So don't worry if you don't see anything happening there. Just type in your password and press enter. Again, all of this text here is coming from our workshop servers. So it says system restart required. There's nothing to do about that. Just once you see something like this, you're good to go. This will be different for you depending on what your username is. And congratulations, you've logged in to the workshop cluster server.
All right, welcome back. So while that video was playing, we shared a survey in the general channel asking if you have successfully logged into the workshop cluster server. And it looks like many of you have answered. We're still waiting on some more responses. So please let us know yes or no, and then we will help you, the folks who weren't able to log in. And also, if you were successfully able to log in, please lend your experience here, your expertise, and try and help out your fellow peers in your pod. So with that, what I'm going to do is ask the control room to suspend our video feed for just a few minutes while we go ahead and make sure that we get all of our students connected into our server. Thank you. Okay. I have just a few notes and things to kind of go over while we're still working on connecting here. Um, so I just want to remind everyone that we shared a survey. And if you have not answered into that survey yet, that would be really helpful for us to gauge where you're at and if you need assistance still. If you had previously answered no and have since been able to get logged in, then please edit your answer and set it to yes. And then also there was some questions that came in about Secure Shell and the Google Chrome browser and these other things. So I thought maybe I would just take a quick minute to sort of explain this here. So we ask all the attendees following along here to use Google Chrome and the Secure Shell app because it is very easy to install on most computers. And it also provides a very uniform experience for everyone. So if I share my computer screen and show you Secure Shell, it'll look very similar to what Evan or Greg might be showing you, or someone on a Windows computer might be showing you. There is not a strict requirement that you use this. So if you have a favorite SSH client, you're welcome to use that. But this is something that we've found over the years of doing these workshops that it generally works very well, and it allows you to sort of conduct all of your work within a like a single browser. And then also the server that we're connecting to, the workshop server, this is a special server that we built for the attendees of this workshop. So this is not normally how you would use Chime 2. If you were going to be using Chime 2 as part of your day job or as part of your research, you would follow our installation instructions that we've made available at docs.chime2.org. And then you wouldn't necessarily need to use the Google Chrome Secure Shell app. So you are also welcome to use the Google Chrome and Secure Shell app combination to log into your university's cluster as well, or maybe some cluster that's been made available to you. So yeah, there's a lot of options there. Just kind of wanted to rehash what all these things kind of mean and where we're going with it. So if you've successfully logged in, great. Just hold tight for a few minutes before we move on to our next sections. And with that, I think we're going to put the stream on pause again real quick, just so that we can get the rest of the attendees all set up here. So thank you. All right, welcome back. So I think we got just about everyone all situated here. If you have not updated the survey poll response, please do so now. That will really help us keep everything situated here. So while we were helping everyone get logged in, we had a question come in that's wanted to know if we'll have access to the workshop cluster after the lectures, and the answer to that is yes. So these user accounts on the workshop cluster are for you to use between now and the end of the workshop. You are welcome to hop on, poke around, do some small tutorials if you're working through, say, some other tutorials on the QIIME 2 docs. That is what this server is for. We will ask that you not run any real data analysis on here, because this is a shared resource that all 75 of the attendees are all sharing these resources here. So for the sake of for everyone else on there, we just ask that you not put too much load onto it. Yeah, we will have this workshop server up and available until the end of the workshop on the 5th day. At that time, we will shut this down, and these user accounts will no longer log you in here. So we will continue watching the general channel here and any of the pod discussions that got kicked off here, just to make sure that if there's any last minute support requests or anything else that came in, that we get you situated. Before we jump into the next video, though, I just want to share a big thank you from the whole control room. It was really, really awesome to see all of the support that all of the attendees gave each other with helping each other log into the server and so on. And then also, let's just take a quick minute to thank all of the instructors, the whole TA crew that we have on hand here helping out. So thank you. So with that, the next segment that we're going to watch here is about sample metadata. And it is a lecture that has been prepared for us by Justine Debelius. Justine is at the Center for Translational Microbiome Research at the Karolinska Institute in Stockholm, Sweden. And at the end of the lecture here, Justine will be available to provide some question and answer type discussion for us. So with that, let's start up this next video segment.
Hi, my name's Justine Debelius. I'm a postdoctoral researcher at the Center for Translational Microbiome Research at the Karolinska Institute in Stockholm, Sweden, where I study the interaction between the human microbiome and cancer. Today, I want to talk to you about sample metadata, which is some of the most important data in your microbiome study, and it's data you'll likely never sequence. I want to talk to you about what sample metadata is, do you need it, where do you get it, and what are the requirements that are hard requirements, and what are some recommendations for it? So let's start with where it fits into the context of your study. As you're performing a microbiome study, hopefully you started with a really interesting question that you want to answer, and you designed a study that would let you answer that question. You went out and you collected some samples, you sequenced them, maybe you did it in-house, maybe you sent them out for sequencing, and now you're at this workshop where we're going to help you understand how to make a feature table and how to do some statistics to answer that research question. However, your ability to answer that research question and do your important work comes back to having metadata so that you can do those statistics at the end. When you go to collect your metadata, the first most important thing you want to make sure you have is information about your main exposure, the thing that you're really researching. If you're doing a study of soils and location, you want to make sure that you have the longitude and latitude. If you're interested in humans and disease status, you need to know whether or not the person has the disease or is clinically healthy. If you want to look at mice and genotype, you want to make sure that you know what genotype your mice are. Make sure that you have this information. I will be honest and say that I have worked on multiple studies in the last year where the collaborator forgot to collect the information, and it just makes it really hard to actually do the work they want to do. The second thing you want to think about is factors that could affect the microbiome that are either related to that main exposure or that might just affect the community. So, for example, age is a factor that affects the microbiome in humans. So you probably want to collect information about the age of your participants in your disease study, especially if there's a difference in age between the cases and controls. If we think about the soil study, maybe you're going to include information about pH, about salinity, about temperature, because those potentially have an effect on the microbiome, and that could be bigger than the effect of longitude and latitude, and you should know about it. If you're looking at mice, you probably want to think about including information about cage or other things that are going on with the mice. So you just want to make sure you have this information. The third kind of data you want to collect is technical information. How were the samples sequenced? How were they stored? Where were they sequenced? You're going to need this information later, both in terms of publication, but also just in terms of being able to do analysis and account for potential differences there. Speaking of publication and what to include, there's actually a community standard called the MIX-S, or Minimum Information for Any Sequence Standard. This standard is used by common repositories like ENA, where hopefully you'll deposit your sequences when you publish your study. ENA provides information about how the project was conducted, the technical information, but it also gives you some standards for collecting your own information about different environments and how to format that. And the goal with this is both that you can answer questions when you do deposition, but also that you know where your samples came from. And MIX-S has a lot of really nice standardized terminology and ontologies. So when you go to do a meta-analysis at some point in the future, your metadata is comparable and you don't spend 3 weeks trying to standardize different information. When you go to format your metadata for QIIME and for your specific analysis, I think there's often an inclination to start with just having 2 columns, an ID column and a description column. Maybe you decide to have one more column that's, your main exposure or whatever, but you start with 2 columns. And this becomes really hard to analyze statistically because as we do statistics, we need multiple samples per group to be able to look for a distribution in those samples. So instead, I want to recommend that you separate your data into different columns. So here, instead of having the description column, I've got a donor column, a cage column, an individual column, and a day column. I like to format this data as a string that's a little bit more descriptive, because then when I make my figures and stuff, I can look at it and I can know the answer. If this doesn't work for you for whatever reason, I recommend using the Q2 types functionality as you put together your data, and you can specify whether a column is a category, even if you code it as a 0 or a 1, or a number. And this is just useful as you think about how you format your metadata. I also want to mention that within QIIME, there is one additional hard requirement for your metadata, which is that the first column needs to be an ID column, and there is a constrained number of ways you can represent that ID. I recommend looking at the QIIME metadata tutorial to specifically understand that, and you can use QIIME, which is an online plugin for your Google Doc. If you can put your metadata into Google, that can help validate it. The other issue I just want to mention as you're preparing your metadata is that Excel is a really popular way to do it. I like Excel, I recommend it. However, with Excel, you want to be really careful with the formatting. There is a great historic example where somebody went in and checked a bunch of human gene studies and discovered that there were mistakes in it because Excel had changed the gene name to a date in an effort to be helpful. So as you're doing your metadata in Excel, just pay attention to those details and double check it early on. And you can always cast it to text if you look at the cell format menu, but just make sure that as you're recording your metadata, in Excel or in something similar that it's formatting correctly. So let's come back to those questions that we asked at the beginning. So first of all, do you need sample metadata? Yes, absolutely. If you don't have your metadata, you're not going to be able to answer your biological question. You're going to be left with a bunch of sequences, and you're essentially going to be trying to do molecular fortune telling, but like without any additional information. Where do you get your metadata? You should be collecting it at the beginning of your study. You should be thinking about how to format it at the beginning of your study, and you should have it in hand ready to go before you start analyzing your data. And finally, what are the hard requirements for your metadata? Well, QIIME has some hard requirements that you include specific labels for the first column. If you plan to deposit, MIMARC standards are a hard requirement for many of the places you will want to deposit your data upon publication. And then there are some recommendations like making sure that your metadata is in multiple columns that will make it easier to analyze. I hope this talk has been useful for you. I hope you understand a little bit more about metadata, about why you need it, and hopefully you will have amazing metadata in the future. Thank you so much for listening.
All right. Thank you so much for that, Justine. So we are now standing by to do some question and answer for any questions that you might have about Justine's talk here. So with that, I'm just going to briefly introduce Justine again, who's here in the call with us. So if you want to say hello and wave. I don't know if the video is going to switch over without you speaking on Slack or on Skype. So what I'll do is looks like we've got a question here about what's the Google tool that you mentioned in the video for metadata validation?
Sure. So there's this really nice Google tool called QIIME, KEEMEI. And you can use that to take your metadata in. It will check your column name. It will, I think, check the formatting. It will not, however, ensure that your metadata is correct, nor will it ensure that it is set up the way you want for your statistical analysis. So excellent if you want to make sure your metadata will work in QIIME, but unfortunately doing metadata is still, it's not going to solve all your problems.
That is a very good point. Yeah, thank you. Let's see. Anything else coming in here?
So far, it doesn't look like we have any additional questions. Some folks are typing right now, so maybe there's some coming. We can just maybe wait another few seconds.
While we're waiting here, I'll just remind folks, we do have some water cooler channels set up. We've got water cooler 01, 02, and 03. So if there's a point where you're caught up on any of the hands-on stuff and you want to pop into one of these water cooler channels and just say hello and start up a conversation, that is totally acceptable. That is what we're hoping you will do. So let's go back to the...
Sorry.
No, go ahead. Go ahead.
Please come ask us questions about this too if you, at the water cooler or wherever, if you don't want to ask it, where it's read live right now, or if it comes to you later, come back and ask us again, please.
We've got one more question that just came in. I'm going to pass this over to the broadcast.
So the question that came in here is it advisable to have a column with the full sample name, or is it enough to have the fields separate?
So in terms of, when I set up my metadata, you can have a column that is all of the sample information in one. All of the sample information in one hopefully is not your sample identifier. So hopefully you've used something like a qual ID or have a somewhat more random sample identifier because, again, lived experience, your sample identifier will never perfectly line up with your metadata, and you will discover a mistake 3/4 of the way through the analysis, and it will kill you slowly. And you will spend the next 5 years of your life trying to go back and fix that one mistake you made. And so having that description column may be helpful, particularly if it's what comes in from your collaborator. But for doing statistical modeling, for actually understanding your data, you really want the separate columns. And you can always put columns back together, break them apart, recategorize them if you suddenly decide, oh, turquoise and blue and navy are all the same group, and so I should squish them together, which maybe answers your question and maybe doesn't. Sorry.
Well, I think that looks like that is the last of the questions in there for now. We are planning on having a general question and answer session at the end of the day as well. I don't know if Justine will necessarily be on the call then. It might be in the middle of the night, but we'll see.
We'll see.
So thank you so much, Justine, for being here. We really appreciate it. With that, I'm going to switch over to our next video segment here. And this is going to be another hands-on session, like the connecting to the workshop server session. So if for some reason between the last like 15 minutes or so, if you logged out of the workshop server, please log back in. Greg is going to share with us a pre-recorded video on some kind of command line interface basics, a quick refresher to let us know sort of the bare minimum about getting around on the command line and some tools that'll be helpful for you for this workshop. It's not meant to be an exhaustive discussion of using command line tools. So with that, I think we're going to start that video, and then we'll have some time afterwards to do some support and Q&A. Thank you.
Hi, my name is Greg Capareso. In this section of the tutorial, I am going to work through a quick command line refresher. So I'm going into this assuming that you have some command line experience, but maybe not a lot, or maybe it's been a while since you've used the command line. And so we will end up talking about some file system concepts. Specifically, we'll talk about relative and absolute paths, your home directory, and the root directory. We'll then go through a little bit of interactive work using some important built-in commands that are really like the essential need-to-know commands for working on a Linux system. And I'll show you some shortcuts that you can use to make your command line work more efficient. I want to start this by talking about file paths and directory paths. File paths specify the location of a file on your computer, while a directory path specifies the location of a directory on your computer. You may have also heard directories referred to as folders. Those two terms are synonymous, and most people end up going back and forth between using the term folder and the term directory. I may do that myself in this workshop. Either of these path types, so I'll generally call these paths, directory paths or file paths, can be relative, which mean that their meaning is dependent on where you currently are on the file system, or they can be absolute, meaning that their meaning is not relative to where you are on the file system. They're rather fully specified with respect to the root of the file system. Now, the root of the file system, otherwise known as the root directory, is a special directory that contains all of the other files and directories on the computer. The root directory is specified by having a slash at the beginning of a path. And so if you see a path like the ones that I have below that start with a slash, that means that they are absolute file paths. So I've got 3 listed here. The first one is specifying the root directory itself. The second one is specifying a special directory. This would be my home directory on the file system. And so that would be /home/Greg. The last one is a file path that is underneath my home directory. So it's under /home/Greg. And this is a directory where I store photos of my chickens. I have one in there called Barbara.png. Barbara is one of my chickens. And so that would be a photo of her. So I mentioned the location specified by an absolute path doesn't change meaning regardless of where you are on the file system. On the other hand, a relative path does change meaning depending on where you are on the file system. And so here you see 2 relative paths. The first one is chicken photos. That's a directory path. The second one is chicken photos /Barbara.png. That's a file path. Notice that these do not start with a slash. And so that is a big hint to you that these are relative paths, not absolute paths. Now, I mentioned my home directory a couple of times. This is an important directory on the file system. This is generally where you would store your files. So like you saw, that was where I store my photos of chickens. And it's also the directory that you'll be in when you log into the server. So on Linux-based systems, the absolute path to your home directory is /home/username, where username is your name on the system. And so for me, that might be Greg. Your home directory is also abbreviated in a few ways that your computer will understand. First, you'll often see it abbreviated as the tilde or squiggle, and you may also see it abbreviated as $home, all capital letters. Since these are abbreviations of an absolute file path, they work like absolute file paths, even though they don't start with a slash. What I'd like to do now is work through some important built-in commands that you would find on any Linux system. Since this is just intended to be a refresher, I'm just going to cover 4 of the commands that are on this list right now. But I'm providing these so that you can use that as a point of additional study. And so if you'd like to develop some really powerful skills working on a command line, you can do that getting started with just the commands that are in this list. At the end of this session, I'll provide a few resources that can help you with your next steps. The commands that we're going to cover here are pwd, ls, cd, and mkdir. Now, I just want to take a quick minute to talk about the anatomy of a command. So when we look at the command ls, which lists the contents of a directory, we can either provide that on its own, as you see this first form. We could provide some options to it. And so when you provide options, you would do that by either specifying a single dash or a double dash, depending on the command that you're using and the option that you're providing. And those options are going to change the behavior of that program or that command in some way. Some commands may also take an argument, which is effectively something that you're providing as input to that command. And we'll take a look at this. What I'm going to do now is I'm going to switch over to my Google Chrome browser, and I'm going to type Chrome://apps. That will give me this screen here where I can see my Secure Shell apps, my Secure Shell app, and some other apps that I have installed here. I'm going to click on Secure Shell for now. This will bring you to a window that looks like what I have up here. You may have already established your connection. This is the first time that I'm logging into this workshop server, so I'm going to enter my username and hostname now. These are information that we have already provided to you. My username is astute reindeer. And the host name, which will be the same for you, is workshopserver.chime2.org. And then I'll come over and hit enter. So I just hit a bit of a snag here, and this is something that I've run into before. The first time that I enter my username and host name, the Secure Shell app wants me to click in this box up here, which is currently highlighted in red. And what that does, I believe, is it adds this as a new connection. And so I was not able to actually connect until I clicked in that box. Now I can hit enter, and I'm prompted for my password. I'm now logged into the system. The first thing that I'm going to do is I'm going to type clear, which just gets rid of any information that is currently in this terminal window. What you can see now is I have my command prompt, and the command prompt is this $ symbol, and what that means is the shell, which is the program that I'm interacting with here, is waiting for me to give it a command, for me to tell it what to do. The command prompt that we have set up on the workshop server is a pretty fancy one. It's a 2-line prompt where you can see on the first line, it's showing what our current working directory is. So that means that this is the directory that we are currently in on the file system. Now, I mentioned earlier that you will typically start in your home directory. That's what's happened here. On this server, my home directory is /mnt/home/astute reindeer. Again, astute reindeer is my username on this system. The first of the commands that I wanted to show you is pwd. So if I type pwd here, that stands for print working directory, or in other words, print current directory. So if I type that, you'll see that that is printing that one line to the screen. It's printing the directory that I'm currently in, and then I get my command prompt back. The next command I want to show you here is ls, which will list the contents of the current directory. So if I type ls, you can see that right now I have one directory here, and that is called workshop. If I wanted to, I could also look at the contents of workshop, and the way that I would do that is by providing workshop as an argument to the ls command. So I could type ls workshop, and at the moment I don't have any files in that directory, and so I won't get anything back as output. Now I want to show you a quick tip here that will save you some typing on the command line. If I were to type lswo, what I could do is I could hit the tab key, and that will auto-complete the name of the workshop directory. The reason that works is because there was only one either file or directory in my current directory that started with wo, and so the shell is able to tell what I mean if I just type those two letters. And so if I type, or if I hit enter now, I'll get the same thing. It'll just show me there's nothing in that directory. The next command I want to show you is cd. And so if I type cd workshop, that is going to change my working directory. Now, with this fancy command prompt that we have here, you can already see that that's been changed. So now you can see that I'm in the workshop directory. If I didn't have that fancy command prompt, I could just type pwd, and that would tell me that I'm now in mount, home, astute reindeer, workshop. If I wanted to change directories back to my home directory, a shortcut for doing that is the cd command. So regardless of where I am, cd is always going to take me back home. So we've now seen how cd works if you provide it with an argument, and that happened when I provided cd workshop above. And you can see what happens when I don't provide an argument. So if I do cd with nothing after it, it takes me just back to my home directory. So I'm going to cd back to my workshop directory, and I'm just going to type cdw and then hit the tab key. And you can see my working directory has now changed to Mount Home Astute Reindeer Workshop. The last command I want to show you in this tutorial is mkdir. This stands for make directory, and this is a command that requires that I provided an argument. And what I am required to provide is the name of the directory that I want to create. And so just to set us up for the next section of the workshop, I'm going to say mkdir mouse_tutorial. And notice that when I create that directory, I don't necessarily see anything happen. I have to type ls. And when I type ls, I can see that mouse tutorial now exists. Because that's a directory, I can change into it. So I typed cd mo, and then I hit the tab key, and that auto-completed. And you can see that my working directory has now changed. I'm now in Mount Home Astute Reindeer Workshop Mouse Tutorial. If I want to go back to my home directory now, I can type cd. If I wanted to, say, change directly into that mouse tutorial directory, I could use command tab completion to help me with that. I could say cd workshop, and that'll be auto-completed. And then I could type m, hit tab, and that would auto-complete. That'll put me in that workshop mouse tutorial directory. Another handy shortcut for using cd is if I type cd ... What that means is I want to change one directory up from where I currently am. And so if I type cd ... you can see that I changed to my Mount Home Astute Reindeer Workshop directory. If I type cd ... again, that will now, this time, bring me to my home directory, Mount Home Astute Reindeer. The last thing that I want to show you here is the use of the up and the down arrow to scroll through my history. So if I hit the up arrow, you can see that I can start scrolling through all of the commands that I've run so far. And so I'm just now hitting the down arrow, and I can run through all of the commands that I've previously ran. If I hit enter, when I've landed on this cd workshop mouse tutorial, that will allow me to rerun that command. And so you can see my working directory changed to workshop mouse_tutorial. I'm going to type cd to get back to my home directory. And that is everything I want to show you in this section. And briefly, I just want to mention a few of the more advanced commands that we'll use in this workshop. In this workshop, we will be using curl or wget. We might use these interchangeably. And what these commands do is they download a file from the internet given its location. And so if you provide a file's URL, you can use one of these commands to download that to the server that you're currently connected to. The other command that we'll spend a lot of time with here is the chime command, and that allows you to use the Chime2 program through its command line interface. That brings us to the end of the command line refresher section of the workshop. I want to end this by pointing you at some resources for practicing or learning more. A great resource for this is the carpentries, including software carpentry and data carpentry. They have many lessons on their website. A very relevant one for this would be their Unix shell lesson. And they also are regularly teaching lessons, or they're regularly teaching workshops around the world where you can learn data management skills and programming skills and command line skills. Another resource is Jocelyn Lee's lesson that she developed for a QIIME 2 workshop a couple of years ago. This works through the commands that I provided earlier, and which I suggested would be a great initial set of commands for you to learn to begin very effectively working with command line software. Okay, thank you very much, and I look forward to working with you later in the workshop.
Welcome back, everyone. We had a great question come in while that video was playing. Someone wanted to know, are there any differences between curl and wget? These are two different command line tools. I've got Greg standing by here. Greg, do you have anything to say about that question?
Yep, thanks for the question. So curl and wget are two different programs or commands that more or less do the same thing. There's not really, as far, for basic usage, there's probably not any reason to really strongly prefer one over the other. Some systems may have one installed and not the other, and so that's why we typically, when we get to the workshop, we will provide options for both. So we'll always provide a command with curl, and we'll provide a curl command, and we'll provide a wget command. I think curl may have some more features than wget does. So if you were gonna learn one of those, curl might be the one to learn. Sounds like Evan prefers wget. There's really not much of a difference, though, that you need to worry about here between the two of those.
Thanks, Greg. I'll also add into that. So many of us pronounce it wget, but there are other pronunciations out there on the internet like weget. So if you hear some of our instructors saying weget, they're referring to the same tool as wget, wget. So yeah, just something to keep in mind. It looks like maybe another question is coming in here. I'm just waiting on Slack. Quick Slack update. It looks like they are still suffering from all kinds of performance issues. So hopefully that is sorted out by the end of the day. Such is life. Just waiting here. So Greg, I'll ask you, how did you kind of get started with using the like a command line interface or running commands this way? What was your path to that?
Sure. Yeah. So my path to that. So when I started, so I have a degree in computer science. And that was really where I got started in this, but it was not where I started in college. So I actually started college in a pretty different field. I was accepted as an art major and got interested in computers and, wanted to learn how to do this stuff. And I took a computer science 1 class where we were doing some C++ programming on Windows, pretty graphical interface focused. And so, that was a good intro. And then when I got to computer science 2, which is data structures and often is thought of as sort of the weed out class for the computer science major, that's where I first started using command line tools. And I'll never forget my first time going into the data structures lab and sitting down and opening up a computer and getting a command prompt. I was terrified. I didn't know what to do. I had, a little bit of information on what to do, but I distinctly remember thinking, this is it for me. I've done what I can in computers and there's no way I'm going to get past this point. And so, I understand the feeling that a lot of you might have right now if you're scared sitting at that command line, because I remember having that when I first got started.
Well, thanks, Greg. Looks like we have another question that came in here. Do these commands work on Mac OS and Windows? And I'll just add to that as well, and Linux. Can we talk about those three options, Mac OS, Windows, and Linux?
Yeah, so these generally these commands should work on Linux and Mac OS, and also, Matt, correct me if I'm wrong, on the Linux subsystem for Windows. They will not. They will probably not work at like the traditional command prompt in Windows. You may observe some differences in how they function on, say, Mac OS versus Linux. But like these core commands, the ones that we just covered, are going to be pretty similar, whether you're on Mac OS, Linux, or Windows subsystem, Linux subsystem for Windows.
Great, thank you. Yeah, we recently added installation instructions to our QIIME 2 documentation about how to install QIIME 2 on this Windows subsystem for Linux. We're pretty excited to see this because it opens up a lot of software possibilities for people that are using Windows computers. A lot of scientific and academic computing tools are usually Linux-based tools, and so getting really easy access to that in Windows is something pretty exciting for us.
Yeah, and just following on that, Matt, I noticed that about 50% of our survey respondents said that they primarily work on a Windows machine. So just another reason why we're excited that we're able to support that finally.
Yeah. Yeah, and it's worth pointing out here, the workshop server that everyone is using is a Linux server. So all the commands that you are running by running them in the Google Chrome Secure Shell app, these are all Linux machines that you're working on specifically. So it looks like that might be it for questions here. So how about we move on to our next segment, which is break time. I think we've got a 30-minute break scheduled. It looks like we're breaking for that 5 minutes early. So please come back here in 35 minutes. We should be on the same YouTube stream. But if for some reason that changes, we will let you know in the general channel. And otherwise, feel free to take advantage of the water cooler sessions. Let us know where you're from. Hop in there and tell a joke or share some poetry or something. Yeah, with that, thank you. We'll be back in 35. All right, welcome back, everybody. Hopefully you get a chance to get up and stretch and maybe eat something. We are getting ready to start our first section of our interactive tutorial for this workshop. So we've kind of mentioned and hinted at this a little bit so far, but we're going to be working with the a tutorial, a QIIME 2 tutorial that we have available on our QIIME 2 docs called the Parkinson's disease mouse study. And we're going to start off with a short video from Justine Debelius from the Karolinska Institute. And Justine is going to walk us through some of the study background and the experiment design, and sort of explain to us what has been done to prepare this real-world data set for the tutorial itself. So there's some subsampling and some other things. So no need to have your secure shell session open just yet, but right after Justine's session, then we're going to start running our first QIIME 2 commands. So with that, I am going to ask the control room to switch over to Justine's video, and then we'll have Justine standing by at the end for Q&A. So thank you.
Now I'd like to introduce the tutorial data that we're going to be working with in this workshop. We're going to work with a Parkinson's mouse tutorial, which is a fairly comprehensive workflow tutorial that walks you through some steps you might take in a standard microbiome analysis, or at least a good place to start with your analysis so you can see what's going on and then decide how to move forward. You may have heard of Parkinson's disease in the context of someone like Muhammad Ali and Michael J. Fox. In fact, Parkinson's disease is the second most common neurodegenerative disorder, and it affects about 1% of adults over the age of 60. Parkinson's, as a neurodegenerative disorder, affects a region of the brain called the substantia nigra. It affects about 1 million Americans, with most diagnosed after the age of 50. And Parkinson's disease is associated with motor symptoms. You probably think of tremors and slow movements, but it's also associated with non-motor symptoms like constipation. One of the reasons that people started to think that the microbiome might be associated with Parkinson's disease was because of the constipation. In 2014, the first article looking at the relationship between Parkinson's disease and the microbiome was published. 2 additional articles followed shortly thereafter, and it was relatively strong evidence for the relationship between the microbiome and Parkinson's disease at some point in the progression. However, all 3 of these studies are case control studies, and so they compare healthy controls to people with Parkinson's disease. And while we can see an association, we can't understand the temporality. And so there are 2 ways that we might see a microbiome associated with Parkinson's disease in these patients. One is that you start out with healthy aging and a healthy microbiome, and then something about your microbiome changes and you develop Parkinson's disease. Or you might see healthy aging and a healthy microbiome, and then you develop Parkinson's disease and your microbiome changes in response to the disease. And so these are 2 possible ways that we could see a Parkinson's disease phenotype in an individual. However, it's challenging to test these 2 hypotheses in humans. We don't know when Parkinson's starts. There's a prodromal pre-symptomatic period that can last for many years, and so we don't know when people transition from healthy aging into that Parkinson's phenotype. Well, it's relatively common, affecting 1% of the population. If you wanted to get a lot of people with Parkinson's disease in a prospective study, starting from when they're in their 40s and following them up into their 70s, you would have to collect a lot of people to be able to get a large enough sample size if you're assuming 1% of the population is going to be affected. It would take a long time to do this. You might need 30 years of follow-up to be able to make this determination. And so it really isn't practical to do this experiment if you want to go further and understand the microbiome can affect whether or not someone develops Parkinson's disease. Luckily, there's a model system that helps us test for this. So there's this idea of notobiotic or germ-free mice. So you have mice that live in a bubble that have no bacteria, and you can introduce new bacteria to these mice. And so you can see what the effect of introducing different kinds of bacteria are. You could introduce a single bacteria. You could introduce a community of bacteria. And so these act as a nice test system for being able to understand what's happening. There is also a mouse model of Parkinson's disease that gives you a susceptible model where it takes the gene that is overexpressed in Parkinson's disease, or a gene that is associated with Parkinson's disease, and it overexpresses the gene. And this leads to a Parkinson's-like phenotype that can then be used in studies. Using this mouse model and notobiotic mice, Sampson et al. Decided to look at what the relationship was between the gut microbiome and the development of Parkinson's-like symptoms in these susceptible animals. And so they did several experiments to get there. They started with just germ-free mice to look at what the effect of any bacteria was. Original experiment, they took 6 healthy donors, 6 with Parkinson's disease, and they transplanted them either into the healthy mice or the susceptible mice. And they had 3 cages with different types of mice, and then they followed them over time and looked at the change in the microbiome and the development of symptoms. And in the original experiment, they concluded that there was a relationship between the microbiome and the genotype, as well as a relationship between the mouse microbiome and the donor. And so in our tutorial, we're going to work with a subset of this data from 2 donors, one healthy, one with Parkinson's disease, and look at the 6 mice that every donor has over 4 time points. And we're going to try to see if we can find a difference in the microbiome associated with genotype.
Welcome back. I have Justine standing by here with me to potentially answer any questions that you might have about this the synopsis of the study, and in particular the study design. So we will we'll just wait a minute here while we give everyone a chance to think about their questions that they might have. In the meantime, I'll ask Justine, how did you get started with like command line computing or, scientific computing?
I was doing a master's in cell biology, and we kept doing actually MATLAB analysis of data. And I realized that I really liked MATLAB and I really didn't like cell culture. And then I started my PhD and I started rotating through labs, and it took me 18 times to do a PCR run. And then I went and I worked in the computational lab, and I could do it. And I switched over and occasionally look back and go, I'm really glad I do computational biology now.
That is fantastic. We have a question that just came in. Someone was wondering if you could sort of just briefly repeat the last part of the experimental design, maybe just summarize the experimental design really quick, like in a minute.
Sure. So I'm not actually quite sure where we are with the videos. So the experiment for this is that we took 6 people with Parkinson's disease and 6 healthy controls, and we took their feces and we put them into a set of germ-free mice. And one set of those germ-free mice were susceptible to Parkinson's disease. They have a mutation, which means that no matter what you do, they will get a Parkinson's phenotype. And then we have a set of wild-type mice who should not develop the same Parkinson's phenotype, should just sort of age naturally. And so the hypothesis for the overall study was to see if we see more Parkinson's disease, if we added stool from somebody with Parkinson's disease to mice that were susceptible to Parkinson's disease. For the tutorial, we're working with a smaller subset of that data. We've only got one person with Parkinson's disease and one healthy control. It shrinks the sample size to a much more computable, manageable level. And so it becomes a little bit artificial. But one of the questions is, where will your donor effect end up versus your genetic effect? And that hopefully simulates some of what the original analysis was intending to do.
Perfect. Thank you so much for the recap there. So ideally, over the next couple of days, working through this Parkinson's mouse disease tutorial, we will get a chance to explore that hypothesis in a bit more detail. Let's see. Greg, are there any other questions?
Yep, I just shared one more on broadcast.
Oh, thank you. So someone wants to know what region was used for the study.
Sure. So this was a 16S rRNA study. We use the 515-806 EMP primers. And I actually think it was processed with the EMP protocol on a robot. It was done in 2016 out of Rod Knight's lab. So EMP 515-806. I think 2x150.
Perfect. Thank you so much. It looks like that is maybe our last question for now. So thank you, Justine, for stopping by and filling us in on some of this info. Thank you. So with that, I think we're actually going to start working with this tutorial data set. So we have our QIIME commands that we're going to run in the next video segment. So that video segment was produced by Andrew Sanchez, a QIIME core developer. He's part of the Capareso lab. And Andrew is going to walk us through some first steps with this analysis that have to do with sample metadata. Feel free to ask questions in general as we're running through the commands. And then at the end of the video, we will take some time to make sure that everyone gets a chance to catch up and so on. But if you have not logged into the workshop server since coming back from the break, now would be a good time to make sure that you're still logged in and that you're set up, because we're going to start actually running these commands and looking at some QIIME results. Thank you.
Hi. In this tutorial, we're going to cover sample metadata, the key to gaining biological insight from your data. And this is quite a large topic. So to begin, I want to show you how you can dive deeper into this topic, because in this tutorial, we're just going to cover the basics that are required just enough to get you through this tutorial. So if we open a new tab and go over to docs.qiime2.org, you'll see on the left here we have tutorials. Click there, and then you can scroll down to find the metadata tutorial. Metadata in QIIME2. So this tutorial, this is going to be the deep dive version of this tutorial. So we'll cover some of the topics that are mentioned here on this page, like formatting requirements, validation, and comments, and identifier columns. But if you really want to dive deep and really understand this topic deeply, which I highly recommend, go over to the metadata tutorial afterwards when you have time. So then what will we learn? In the previous lecture, you went over metadata from a more high-level theoretical perspective. And in this tutorial, we're going to cover the sort of nuts and bolts on the command line. So understanding metadata in QIIME2 and how to inspect metadata on the command line and the relevant commands involved in that. Not just the commands that we went over in the command line interface refresher, but QIIME2 specific commands for metadata. We're also going to go over metadata column types. QIIME2 has a somewhat of a special format for its metadata, and we'll learn what that is and how to take advantage of it and why it's important. And we're also going to look at viewing QIIME2 artifacts, which end in .qza, and QIIME2 visualizations, which end in .qzv. And you will see that in throughout the rest of these tutorials. We're also going to go over the general workflow that you'll use throughout the rest of these command line tutorials. So that includes copying and pasting and then running those QIIME2 commands in the Secure Shell app. Also getting a link to the output. So a lot of these commands are going to give you what we call artifacts or visualizations, which I just mentioned. And then we're also going to cover how to navigate to a website called view.qiime2.org. And that's going to allow us to view the data, visualizations, and provenance, which I will discuss in more detail. Before we get started, let's go ahead and briefly review how to log into the workshop server using the Secure Shell app. If you've already done this, then don't worry about it. You just be patient as we go through this step one more time. We have a lot of different skill levels in this workshop, so we just want to take it slow at the beginning and make sure that we're all on the same page. So first step is to type in the URL chrome://apps, enter, and find the Secure Shell app icon here. Click on it, and if you've already logged in, all of this information is going to be pre-filled. But let's do it again one more time. So type in your unique username and workshop-server.qiime2.org and press enter. Type in your unique password, and again, nothing is going to show up here. That's perfectly normal. Enter, and we're in. I like to clear the screen at the beginning, and it's always nice to take a peek and see what we have here. Should be, mine's empty, and unless you've created something, it should be empty as well. And we need to change into our workshop directory, and then one more, change directories into the mouse tutorial directory that you should have created in the CLI refresher tutorial. And with that, let's get going with sample metadata. So let's get started by going through an example of this general workflow by heading over to docs.qiime2.org. Here on the left, you'll see the table of contents, and down below, tutorials. Go ahead and click tutorials. That's going to expand to give you more options, and if you scroll down, you will see the Parkinson's mouse tutorial. From here, scroll down to the metadata section. Now let's copy our first command. You'll remember the first step in our general workflow is to copy a command. And the first command is downloading the metadata that we're going to be working with. So typically, if you're just working on your own computer, you would click this link and it would download the file here. And you could open that and view it on your own computer. So this is the file, this is what the file looks like, and I'm just viewing it here locally on my laptop, on my MacBook. However, we're going to need this. I'm going to exit out of here. We're going to need this on the workshop server. And that's what these tabs here are for. wget and curl are the name of Linux utilities. We're going to go with wget, which is the first option here, just to make sure we're on the same page. And this does the same thing, but it will, when you run the command on the Secure Shell app, it'll download it to the workshop server. So let's go ahead and copy by clicking this clipboard icon and coming over here to the Secure Shell app, and you can paste it into the terminal here. And on a MacBook, it's Command V. On a Windows machine, I believe it's Control V. And here it's telling us the wget is going to output, capital O for output, the file to this file metadata, a file named metadata.tsv. And the last part of the command is just the URL where that file is located on the internet. So press enter, You're going to get a bunch of nasty output from wget. You can pretty much ignore that, except for the last part. You can see here it says that metadata.tsv was in fact saved. I'm going to clear my screen to get rid of all that stuff. If I press ls, I can see that I do in fact have the metadata file. And the file is located in my working directory, which you'll always have up here in the Secure Shell app. This is a unique feature of the Secure Shell app, which is kind of convenient. And the important part here is that you are in the home directory named after your username and in the workshop directory. That's going to be very important moving forward, and I will show you why. You'll remember from this slide on the workflow that the second step is to get a link to the output. And by output, I mean the output of the command that we just ran, in this case, which was metadata.tsv. So in order to get the link to that, we're going to open a new tab. We need to navigate to a URL, workshop-server.chime2.org / your username. In my case, Zippy Oxalotl. Make sure you type in your unique username. If I press enter, this is actually going to show me the contents of that workshop directory. So if I go back over to the Secure Shell app, I can, if I move into the workshop directory by typing cd ... to move one directory back, and at ls, I can see the contents of this workshop directory is a folder, mouse tutorial. And that's where I just was. So I'm going to jump back into there by typing cd-. That's a special command to go back to where I just was. Now if I go back over here, again, this is the workshop directory. And then, so what's happening here is I'm viewing the files that are in the workshop server from my browser, as opposed to the Secure Shell app. So if I click in this mouse tutorial directory, I can see the metadata file. Now what's going to happen if I click on here is it's just going to download the metadata.tsv file. And I'm going to come back to my slides real quick because the next step was to view the data at view.chime2.org. I'll show you if I open a new tab and go over there, view.chime2.org, you'll see that this is meant for viewing .qzas and .qzv files, artifacts or visualizations, directly from our browser. So there's one step that we're missing here because we have a metadata.tsv file. And what we need is a .qza or a .qzv. So let's come back over here to the tutorial and see what's next. So the next step is another chime2 command, and we're going to copy it just like before by clicking the clipboard. And coming back over here to the Secure Shell app, I'm going to paste the command, press enter. So chime metadata tabulate. This command takes a input file, which is a tab delimited format, and it outputs a visualization. So this is the .qzv file that we needed. And just one quick tangent about chime2 commands. We use a convention on the chime2 command line interface that you'll see throughout the workshop, and that is to prefix commands with a meaningful letter. So this -m, you might be able to guess, --m means metadata. So that's telling us that this input file needs to be metadata. And this --o, that stands for output. So you'll see these prefixes on a lot of commands throughout the tutorial. And I'll show you a couple more in the next ones. So we have this visualization, metadata.qzv. Now if I come back over here to my workshop server.chime2.org /username URL, and I refresh, it's going to refresh the contents of what's now in the directory after we've run that command. In order to get the link to this URL, simply right-click and click on Copy the link address. Now, on my clipboard, I'm going to have the URL that is needed by view.chime2.org. So, down here, it says I can provide a link to the file from the web. I'm going to click there, and that's going to give me the option to paste in. So I can right-click paste or Command-V, Control-V, and this is my unique link. You can tell it's unique because that's my username to my metadata Chime visualization file that I just generated with the Chime metadata tabulate command. So let's click go and see what happens. So what we have here is essentially a spreadsheet, but in Chime's specific visualization format, which is being viewed using view.chime2.org. Now, you'll see that just like a normal spreadsheet, we have column names up here at the top and rows with values for each of those columns. The difference here is a few things. One, we can sort this spreadsheet using these buttons here, or sort the visualization rather. And one thing to note about that is that you can't really mess this up because this is a view-only format. So we can't change any values in here. I'm clicking, I can't change anything. When I sort, there's no way of messing up the sorting, which you may have done in Excel or Google Sheets before. The other notable thing is that in addition to the rows and the column names, we also have an additional row here. And this leads me into my next topic, which is all about types in Chime2 metadata. So in addition to the normal parts of a spreadsheet that you're probably familiar with, we also, as I mentioned, have types, column types in Chime2 metadata. So Chime2 currently supports two types of metadata columns. That's categorical and numeric. Jumping back over to the visualization, you will see we have mostly categorical and numeric. And from here, it might be a little difficult to tell the difference because you can see a numeric column here. It all has numbers, yet this categorical column has numbers as well. So what's the difference? What gives? Well, Chime2 has a heuristic for inferring the column type. So if the column has only numbers or missing data, then Chime2 will make a best guess that it's numeric data. If the column contains any non-numeric values like letters, then it's going to guess that it's categorical. Missing data are supported in both categorical and numeric columns. So you can see here, going back to this example that I just mentioned, because Chime2 would have guessed that this column is numeric because it only contains integers, as well as this column, we need to explicitly tell Chime2 that this is categorical. And the way that we do that is with comments, basically. So if I come back over here to the Secure Shell app, I'm going to take a peek with the less command into this metadata.tsv file. Remember, this is the original file that we started with to generate that visualization. So I want to make sure that I create the link between this file and the resulting visualization. So here we have, you'll notice that all of these column names match up. It's really just the same exact information that's in here. This is just a pretty version of it, basically, and read-only. And under here we have these type declarations. So the first one is a comment. It starts out with a hashtag, and it's just telling Chime2 that this line is going to contain our types, our metadata column types. And here, all of these are metadata column types, and they correspond to what we have here. And again, This is very important because it allows us to explicitly specify the types of these metadata columns. And the reason that's important is because while this mouse ID isn't, they are in fact numbers, we need QIIME2 to know that it's actually categorical metadata. So for example, we're not going to be doing any math on these numbers. And they are categorical in that while there is a infinite range of numbers between 547 and 546, say, there is no mouse ID that is 547.2, for example. So it's very important that we explicitly specify that metadata. And that has the advantage of our metadata. It makes our file more descriptive and unambiguous. So this comment directive is very important because it allows us to explicitly state the column type and avoid the column type inference, which I discussed previously up here. So again, as we discussed with the mouse ID example, it's useful if apparently numeric data should be treated as categorical. Another example, a subject column where subjects are labeled 1, 2, 3. So just to go in a little more detail about how this comes into practice, it really comes into play when we're talking about doing statistics on our with our metadata. So numeric data is a continuous variable. We can do math on it, and that means there's an uncountable set of values. So numeric data, we can do correlation tests, whereas with categorical data, we can't do correlation tests. But we can do other types of statistics like group significant tests. And categorical data, another way of describing it is that it's discrete variables, finite permitted values. So that's all for the introduction to metadata in QIIME2 for this workshop. Remember, this is there's a whole lot more to this topic, so if you want to really dig in, head over to the documentation homepage at docs.qiime2.org. Scroll down underneath tutorials and you'll find more information in the metadata in QIIME2 tutorial.
Welcome back. We shared a poll in the general channel asking folks about where they made it in that set of the tutorial commands. If you didn't get a chance to answer the poll, please do so now. We'll use that to kind of gauge where everyone is at and then help provide support to them there. So if you have general questions that you'd like to discuss, now would be a great time to post those into the general channel. Otherwise, if you're stuck, if you ran into issues, maybe you had an error message or something, let us know in the general channel, and then we will get an instructor to help you over in your pod. So we'd like to move as much of the support discussion into the pods as possible, since as you saw earlier today, those can involve quite a lot of traffic and discussion and whatnot. So, and if you didn't make it all the way to viewing the metadata file at Q2View, that's okay. We'll work with you to get there. So while folks are answering that question, I will introduce the creator of that video. So I have here with me Andrew Sanchez, and Andrew is a research software engineer in the Capareso lab at Northern Arizona University. And so, yeah, Andrew, welcome.
Howdy. Thanks, Matt. Happy to be here. This is my first QIIME2 workshop, actually. Just happened to coincide with the first online workshop. Really excited to be here, and I just joined the team earlier this year, and it's been awesome.
That's great. So I've got a question here that came in, and someone wants to know. Let me just double check that I'm in the right channel here. When is it necessary to include a barcodes column in your metadata file?
So I think this is getting into what we're going to be covering coming up in one of the next tutorials. This is getting into the topic of multiplexed data and how to demultiplex that with QIIME 2. So that's the short answer. Do you guys have anything to add to that?
Greg, it looks like you've got something to add.
Yeah, I'm happy to add just real quick. Like Andrew said, that's something we'll cover in one of the next sessions. But basically, the idea is like if your sequencing center has not demultiplexed the data for you, then you'll need to include barcodes. If you have, if they have been demultiplexed for you, then you don't need that information. If those terms that I'm using, like demultiplex versus not demultiplex, don't make sense to you yet, they will soon. We'll be covering that in one of the upcoming lectures.
Thanks, Greg. So, Greg, are there any required columns for QIIME 2 metadata?
That's a great question. So the only column that is actually required for QIIME 2 metadata is the sample ID column. And so the first column of your metadata file can be named one of a few different things. The document that Andrew pointed you at in his video, the metadata tutorial document, covers those. They can be called things like ID or sample ID. There's a few variants on this, but that is the only column that is absolutely required for QIIME 2 to work. Now, going back to Justine's lecture, if you want to do anything interesting with your data, you're going to need more information in there. But as far as QIIME is concerned, you can get by with just that column.
We have another broadcast question that just came in here. So someone would like to know, how can we tell QIIME 2 if a column is numeric or categorical? And Andrew or Greg, interested in answering that?
Sure. Yeah, so I think I touched upon it maybe briefly, but if you look inside that metadata file, you'll notice on the second line starts with a hashtag, and there you have the metadata column types in this second sort of special line that QIIME 2 understands. And that's how you can define, you can sort of annotate the metadata file with extra information about each column type. And we currently only support two different types, which are what you see there, numeric and categorical. And again, if you go to the docs.qiime2.org website and find the metadata tutorial, reading through that probably a couple times is going to behoove you in your endeavors with QIIME 2.
Andrew, how would you go about making a QIIME 2 metadata file? Like, what tools would you use? Would you use like Google Sheets or Excel or?
Great question. I've myself have never actually handcrafted one. I think you could use a variety of tools. Maybe Greg or Matt could dive into that one in more detail.
Yeah,
Great.
I'd be happy to talk about what my normal workflow looks like. So normally, what I'll do is I use Google Sheets most frequently for this, and then from Google Sheets I can export that as a tab-separated text file, and that will be directly what I use with QIIME 2. And the reason I like using Google Sheets for this is that often I will be working with multiple people on creating that sample metadata file. And so, for example, I might create the set of sample IDs using a tool like QualID, which came up earlier in the day. I will then share that with some people who might be out in the field. Actually, I was just collecting soil samples a few weeks ago. Some folks are out in the field collecting these, and so they might be adding some of that information. And then later on, when I ship this, off to the lab, they're going to do things like assign barcodes to samples, and so they could enter that information in the Google Sheet as well. And so if I'm using Google Sheets, then everybody can be collaborating on the same document, and that way we don't lose track of who has the latest version, or if somebody edited version 1 and somebody else edited version 2, and now we have to figure out how to merge that back together. Now, I think you can do a lot of the same stuff with Microsoft Excel. I don't use that as much, so I don't really, I can't really advise on that. But I like that idea of like a cloud-based way of editing this so that everybody can just be accessing one copy of the document. Now, a word of caution. Excel seems to be a little bit more problematic with this, but Google Sheets definitely does the same thing. You have to be extremely careful with auto formatting in these spreadsheet editors. And so you want to be really careful, for example, that some formatting rule is not being applied when you're entering data. So for example, like some kind of a date formatting, or some kind of a rounding or numeric formatting that you might not be expecting. That is a very common way that people get into trouble, is that their spreadsheet editor is making changes to the file that they're not aware of. Just something to be aware of, something to always be careful of.
Yeah, Bod shared a interesting BBC article with the instructors. Maybe Bod, if you could share that in Water Cooler 01, that would be useful. But it's about COVID-19 case misreporting because of Excel-related issues, just like what Greg was talking about there, with like columns being formatted incorrectly and whatnot. So I have another question here, and I'll direct this one towards Greg. So this one's maybe a little bit more of a best practices question. So what do we do if we have a lot of metadata that we're working with? Like, imagine we have very robust study design, data collection program. Is there some kind of limit as to like how much metadata we should include in our QIIME 2 metadata files, or recommendations on how to approach that?
Yeah, so that's a good question. Definitely, the limitations that you're more likely to hit, I think, are with your spreadsheet editor than with QIIME 2. And so when you're dealing with really large metadata files, it's possible that you could get, potentially beyond row or column limits in those files, or that it can get so big that you it just becomes very inconvenient to work with. One good practice here is, there may be some information that doesn't necessarily need to be in your sample metadata file. So if, it's not stuff that you're going to be working with directly in your microbiome analysis, and you're running into those limits, maybe you have a secondary sheet in that spreadsheet editor where you have additional information. And so I've done this before for some of my studies, where I'll have multiple sheets or tabs in a spreadsheet editor like Excel or Google Docs, where I've got sort of the most important information that I am interested in for the microbiome analysis in one sheet, but then I use those same sample IDs in other subsequent sheets. If you have, another thing is, like, if you're doing a temporal study, so like a longitudinal survey where you have a lot of time points, it might also make sense to think about like having sample IDs and also subject IDs. And then you could have a separate sheet that would provide information on each subject, so that you're not duplicating that multiple times in that one sample metadata spreadsheet. Now, QIIME 2 doesn't directly know how to interact with, that kind of scenario, like a secondary sheet that has some additional information. But if that's a situation you're running into, get in touch with us on the QIIME 2 forum, and we can try and help you figure out what's going to be the best way to manage that metadata.
Great, thank you, Greg. I have one more question that I want to direct at Andrew, but before that, I just want to remind our watchers here, if you are stuck and need some help, let us know in the general channel, and someone will be in touch and will organize a call or a discussion in your pod, in your support pod. As well, if for some reason during this workshop, if you get stuck and aren't able to continue working with the interactive commands, another option that is available to you is at the end of each section of commands in the Parkinson's mouse disease tutorial, there are links that you can click on that will allow you to view the visualization. Actually, Evan, can I do a screen share real quick? Let me share my screen. Am I sharing here? Thank you. So I am at the QIIME 2 user docs. So this is docs.qiime2.org. And I'm going to scroll down. Oh, is it sharing my tab? Let me double check this here. My apologies. How about now? Is that a docs? I actually don't have that tab anywhere on my screen. Let me see. Let me just share my whole desktop. How about now? Can you see the user docs? Thank you. Perfect. Okay, so I am at docs.qiime2.org, and I'm opening up the Parkinson's mouse tutorial. So this is the same tutorial that you've been working with in this first command. And if I scroll down to this first set of commands that Andrew had us run, right below the command is a listing that has all the outputs from that particular command. And here we have metadata.qzv, and next to it are two links, view or download. So if you get completely stuck and are not able to move forward, this is always a good option for you. You can come over and click on view, and it will open up a tab with the visualization or the artifact that you're interested in looking at. So I won't go into the details of the content of this visualization, but just in case, as a sort of fallback in case you get stuck. So with that, I'm going to stop my screen share. And I'm also going to make a note to learn how to use Skype at some point. And I have one more question. Andrew, do you have any thoughts on if a sample ID, what the sample ID column should look like? Should it be alphanumeric sample IDs, or what are the QIIME 2 rules there? And then maybe some of the best practices Greg could talk about with that.
Sure. Yeah. Good question. It's important to think ahead about the dirty details like this, which I'm sure you'll quickly find out as you continue. Sorry for not. I don't know if my video was on earlier. I wasn't trying to hide from anybody. I just forgot to turn it on. But so again, that metadata tutorial. This is another example of something that you'll find covered in detail there. It's really parts of it are very good for reference, and this is a great example. So the header the name of that actual ID column has a few requirements, which you can find there. So just briefly, you can call it ID, sample ID, a few other things. It's case sensitive. There's a few rules that you got to keep in mind with this stuff. And then as for the IDs themselves, yes, they alphanumeric, Unicode characters only. There's a few symbols that are allowed, like a period or a dash. They have to be under 36 characters long. At least one ID must be present in the file. IDs can't be empty. They have to be unique. And yeah, just so you get the idea, there are a few rules that you need to keep in mind. And I think that's also where this QIIME plugin for QIIME 2 metadata would come into play. Is that right?
That is, yeah. And I'll just add one little amendment to that. So QIIME 2 metadata supports any Unicode character. So basically any character. That does mean that your metadata could be made up of emojis or pictographs or whatever. But just because you can do that doesn't necessarily mean that you should do that. And so some of those recommendations that Andrew mentioned in there about like the length of sample metadata IDs or including dashes or periods, those are kind of part of the best practice for metadata. And those have to do with different plugins and the tools that those plugins are using underneath the wrapper of the QIIME 2 plugin. Greg, do you have anything you'd like to add about this?
Excuse me. Nope, nothing that I want to add. I was just about to, as you were talking there, I was just about to share a link that defines or that will take you to the specific spot in the metadata tutorial where we talk about what the rules are and what the recommendations are. Yeah.
Great. So what we are going to do now is we're going to put the broadcast on hold for somewhere between 5 and 10 minutes, and we're just going to get all of the instructors available to help make sure that everyone who was trying to run the commands was able to get there. And then at the start of the hour, we will begin a general question and answer session. So yeah, if we could switch over to that pause screen, that would be great. All right, just a quick status update from the control room here. We're wrapping up helping out some folks that ran into some issues while running those commands. And as soon as we are more or less done there, we will start our afternoon's general question and answer session. So hopefully in about 5 minutes or less. Thank you. Welcome back, everybody. We have a few more questions related to this sample metadata section of the tutorial that we will cover right now. And then once we're done with that, we'll move into the last session for the day. And that will be kind of general questions and answers. So things that maybe didn't fit in with earlier sections or segments. So before we do that, though, let's kind of go through these last few questions that came in. So I'm going to direct this question towards Greg here. So we have a question here. How do I deal with numbers in categorical columns? Will that be an issue? Will that be a problem?
Okay, so that's a good question. So numbers in categorical columns, typically what you want to do there is you want to use that Q2 types directive that Andrew talked about and define that as a categorical column. Now, if you have numbers and non-numbers in that column, that's not something you'll need to do. QIIME2 will figure out that not everything in that column is a number, and so it'll call it categorical data. But like the example that Andrew showed, where, let me see, I think it was the mouse ID column. Those were numeric values, but they were not really intended to be interpreted numerically. And so, for example, you wouldn't really run like a correlation test with the mouse ID. It's just not how they're intended to be used. And so in that case, as long as you annotate it as categorical, it'll behave as you would expect. You'll be able to use it, for example, in like group significance tests, and it wouldn't show up, say, as an option for a correlation test.
All right, thank you. Greg, the next question that's in our queue here about the diversity results, do you want to talk about that transition to the general Q&A?
Let me quickly talk about that one now, because I think it might just be a good follow-up to what I was just saying.
Okay.
And so the numerical data in your metadata table, if it's not showing up like in your, so I'm guessing here that like the issue might be that like it's not showing up when you want to use it in a specific test. It's possible, one thing to check for is whether you have missing data in that column. Some of depending on which action it is that you're using in QIIME2, it may or may not be able to handle missing values for columns. And so, you know, if you were missing that pH for 5 of your samples, depending on the action, it may not be able to use pH for that. And so that would be probably the first thing that I would check. The second thing that I would check is just review the information that you have in there and make sure that it is all numeric. And so if you're not specifically annotating it as numeric, and you have some other character, like maybe you have, you know, the letter O instead of a 0 somewhere, QIIME2 might see that and consider that to be a categorical column. A great way to test for that is just to generate that visualization that Andrew showed you, because that one will tell you how each column is interpreted by QIIME2. So you'll see that information in the top of that file.
Great, thank you. So I think that kind of rounds out the last of the questions related to the sample metadata section. So I'd like to transition our discussion now into the general question and answer format. And before we do that, I have an announcement or two. So the first thing that I'd like to just mention is, again, just like we did with yesterday, today, this afternoon, maybe in like an hour or two, we will share in the announcements channel the links for tomorrow's videos. So if you would like to pre-watch any of the video segments, or if you have scheduling conflicts or something, please watch that announcements channel. We will share those video segments there. And then also we have a homework assignment for everyone. At some point between the end of, or between now and the start of the session tomorrow, we would like everyone to jump on over into their pod and say hello to everyone in the group and introduce themselves. You should know which pod you're in based on the username. Hold on just one second. Okay, sorry about that. We should be able to tell which pod you're in based on your username. It should say pod 01 or pod 02 at the start of it. If for some reason you don't see that, or you don't see a pod channel that you've been assigned to, just let us know in the general channel and we'll help you out. But your homework assignment is to hop in there at some point, say hello, introduce yourself, maybe share something about like what you're hoping to learn out of this workshop, or what it is that your role is, or where in the world you're located. Just say hello. And so with that, I would like to start off some of the general Q&A questions. So, by the way, I use the phrase Q&A often. That is shorthand for questions and answers. And we just use that in our Chime 2 workshops to sort of talk about like discussions or helping answer questions that you might have. And so this first question that we have here from Marco is there a way to save some info in the metadata without necessarily putting it in the metadata table? And I think I'd like to ask Greg to jump in and say a few words about that. I think this is related to some things that you brought up earlier in the discussion.
Yep. Yeah, that's a great question. And so it's really common that you might have other information about your study. So for example, co-authors on the study, or the hospital where the study is carried out, or the platform used to generate the data. What I would typically recommend, and this actually aligns with how some of the public repositories are going to want you to deposit your data if you're planning to make all your data publicly available, which I encourage. What I would recommend doing is keep a separate file that is sort of your study metadata. And so think of that information as data about the study. And so it doesn't necessarily need to go in your sample metadata file. It goes in a separate study metadata file. And this is probably not something that you would ever really end up providing to Chime, just because, like, based on what you are suggesting here, it sounds like there's some info that's not really relevant for the Chime analysis that you're trying to manage. We were talking a little bit earlier about what happens if you have too much data for your files. If you end up duplicating a lot of that information, that's a good way to get into that situation where you just have files that are too big to work with in practice.
Great, thanks for that, Greg. So there's another
Real quick,
question.
Also, it looks like we've got a bunch of our instructors online right now. So maybe we could just have everybody say hi real quick and introduce themselves.
So I'll do roll call and ask so that we're not jumping on each other here with saying hello. So does that work?
Yeah, that's great. Thank you.
Oh, yep. So I'll start with Bod. Bod, would you like to say hello, introduce yourself to everyone?
Sure, thanks a lot, Matt. Is my audio okay?
Yeah, it's a little bit quiet.
I will try to raise that after this. Yes, so my name is Merbod Astaki, or I think as most people here call me Bod. I'm a postdoc at the Night Lab at University of California, San Diego, and I've been working with the Chime team for a few years now in various capacities. So happy to take any questions from the user perspective, I guess, not so much the developer side of things.
Great. Thanks, Bod. Mike, are you available to say hello and introduce yourself to everyone?
Yes, I am. Can you hear me?
We can, yeah.
I guess I could turn on my video here, maybe. Sorry, I was dealing with some other things. So Mike Robson, I'm an assistant professor at the University of Arkansas for Biomedical Sciences. And I'm just, all around microbiome, human microbiome work. And you'll be hearing from me later in the workshop about phylogenetics.
Great. Thanks, Mike. Would you like to say hello to everyone?
Hi. My name's Ariel Belk. I'm a PhD student at Colorado State. I work for Jessica Metcalf, who came out of Rob Knight's lab, which is how our weird 6 degrees of separation works here. I've been an instructor for a few Chime workshops we've held locally and internationally. And I'm really excited to talk to you all about, I talked about beta diversity and also the Chime 2 classifier tutorials later. So I'll be around to answer questions for those, and like Bod, more from the user side than the developer side.
Thank you. Ben, are you around? Would you like to wave hello to everyone?
Hello, I will not turn my mic on. Oh, sorry, my camera on. I just woke up. I'm a lecturer at the University of New South Wales in Canberra, Australia. Sorry, I would have been here earlier, but as you can tell, I'm a little bit under the weather today. And I'm currently looking at what's going on Slack, and it looks like you've had an amazing day. I'm just trying to get my head around everything that's going on. I'll try to be online a little earlier for the taxonomic classification session on, I think, your Thursday.
Great. Thanks, Ben. Sorry to hear you're not feeling well.
Oh, it's okay. I'm past the worst of it. And it's not COVID. I got tested.
That's good. That's good. Chloe, would you like to hop on and say hello?
Sure. Can you hear me?
You can, yeah.
Awesome. Well, hello. I'm Chloe. I'm a PhD student at the Capareso lab. Yeah, I don't really know what else to say.
Well, we're really glad to have you here, Chloe. Chris, would you like to say hello to the folks?
Sure, happy to. I'm not sure if my video's up, but regardless, my name is Chris Keefe. I am a master's student in the Capareso's lab, the Capareso lab. I've gotten almost enough sleep the last week, and I'm really excited to talk with you guys about mostly alpha and beta diversity, and a little bit about Q2 longitudinal during the tutorial sections of the workshop.
Great, thanks, Chris. My apologies here. I'm afraid I'm going to mispronounce your name, so please correct me. But Renato, would you like to say hello? And then please correct me, because I'm sure I'm not pronouncing it right, and I apologize.
So hello, are you hearing me?
Yes.
So it's Renato.
Thank you.
So I'm from Brazil. I'm a PhD student and also a bioinformatician at the Valley Institute of Technology in Belém, Pará. I'm Guilherme's student. Guilherme is my advisor in the PhD, and although I'm an instructor, I'm also learning a lot about Chime 2 as well. I used Chime 1 a lot, and I learned about Chime 2, but never had the opportunity to put the hands on. So now I hope to learn a lot about Chime 2 as well.
Great. Well, thank you so much for joining us here this week. Anthony, are you around? Would you like to say hello?
I'm Anthony Smart. I'm an undergrad in Greg Capareso's lab, majoring in computer science at NAU. And that's about it.
Great. Thanks, Anthony. Evan, I don't know if you're around or if you're in class right now, but if you'd like to say hello. Okay. So Evan just let us know that his voice might not be able to come through on the broadcast, but he is around. He is here, and he says hello, everyone. And we will get to see Evan later in the week. He's going to give a talk on ANCOM and differential abundance. Did I miss anyone in the call? Okay. If I did, just ping us in the chat and we can circle back on that. So yeah, let's go into a few more general Q&A questions. So there was a question that came in a little while ago about wanting to know if it would be appropriate for students to ask questions about their personal data, or maybe data sets that they're running outside of the QIIME 2 workshop. And I don't think there's anything inherently wrong with that. Just, keep in mind that there's only so many instructors here. And so trying to sort of discuss those sort of specifics is a bit of a balance in terms of serving the whole group versus providing some specific hands-on one-on-one discussion. So if things get into more specifics about your study, then maybe we would talk about having like a water cooler chat during a break or something else. But generally speaking, yeah, ask us questions. If you've run into an issue or trying to figure out how you would run a certain set of commands in QIIME 2 for your study, those are great questions. So just keep those in mind. Anything that you'd like to add, Greg?
And if we are not able to get to it, just go ahead and ask on the forum too. And that's a lot of what we do on the forum, is helping people with their specific, study questions. And those often end up, they usually end up being questions that are more general that, more people can learn from. Okay. So thank you, Greg. Ariel, not to put you on the spot here, but I'm wondering if you would be available to answer a question for us.
I can certainly try.
So we have a question here asking if we need to identify columns in our sample metadata as categorical or numerical before creating our metadata file in QIIME 2. Do you have anything you'd like to say about that?
Sure. So from my experience, you don't have to, because QIIME has the ability to read in and assign whether they're categorical or numerical when you're importing it into QIIME. But if, kind of like we were talking about just a few minutes ago, if you, for example, have numbered categorical instead of giving them letters, like treatment 1 and treatment 2 instead of treatment A and B, QIIME might read those as numerical. So in that case, you would want to include that as in your column header so that it would read them incorrectly. So that way you could do categorical statistics when we get to those in the future.
Great.
Can I answer that?
Yeah, that's perfect. Thank you so much. I'm going to put Bod on the spot next. Bod, are you around?
Yes, sir.
Bod, I'm wondering if I still have primer sequences in my reads, do I have to put the primer sequences in my metadata file? And maybe part of this this answer here also touches on sort of some of the topics that we'll get into tomorrow. I guess just keeping that in mind.
Sure. So the short answer is yes. It's definitely something you want to keep track of as you move towards the next steps, because those primer sequences ideally are removed. So yeah, my short answer is yes, without spoiling the fun for tomorrow. But if you don't plan on removing those, then of course that becomes not necessary.
Great, thank you. Anyone else up here want to add to that at all? Okay. Let's see, who can I put on the spot here? I've got a question. Why can't we run Chime Tools View in the Chrome Secure Shell app? I could put Chris on the spot. Okay, Chris.
Hi, folks. I'm not sure if you can hear me, but I assume you can. Some thumbs up from people. I like it. So Chime Tools View is a really useful tool when you're working from a command line that has the ability to send information to a graphical interface, essentially. Because on the server, we're working in what is called a headless environment. There's no way for that server to display like the graphical information that you would want from a visualization view. And that's why we're using Chime2 or view.chime2.org. It's doing essentially the same thing. It's providing us a little bit more information than Chime Tools View is ever able to, which is cool. And it's allowing us to kind of use that headless environment for our data processing and creating those visualizations, while still letting us view the things that we produce.
Great, thank you, Chris. We have another question that came in. Sorry, my Slack is going crazy here. Another general logistics question here about accessing the videos after the workshop is over. We do have plans releasing all of the videos, the individual video segments, and the live stream broadcast after this workshop. However, we won't be able to make those publicly available until sometime next year. So we're currently planning on doing that in the first quarter of 2020, so sometime between January and March. And the best way to learn about when these videos become available is to sign up and subscribe for notifications on the Chime 2 forum. But that is our intent, is to make these videos available to people at a later date. And let's see. I have another question in here about, let's see, it says, if I'm going to analyze 3 groups of animals, would I create individual metadata files for each of the groups of samples? Mike, would you like to talk about that one? Are you still on the call?
Yes, I am.
And by the way, sorry to interrupt real quick. So the instructors, we're watching the private channel questions broadcast. If you're wanting to reread these after I read them out loud, they're available in questions broadcast.
So this was about the metadata question, correct?
Yeah, the analysis of multiple groups.
Yeah, so usually with the metadata file, I often just annotate, or I have a column, let's say, called treatment group. And in that column, I can define for each individual sample what that treatment group is. And a lot of times, depending on how I plan to use that information in the future, I might have several redundant columns explaining the same information for either purposes of making figures. I could have shorter hand descriptions versus longhand. But yes, the idea, I try to put all of the information associated with the study in one file, or as few files, but usually one file as possible. Hopefully that answers the question.
Great, thanks so much. Yeah, that's what I would do too in that case. Let's see. Any other questions here, Greg?
There was one. I may have just directed it the wrong way. I was supposed to go to questions broadcast. I'm trying to share that again.
While Greg is doing that, I'll ask Chloe a question. Chloe, how did you get started with command line tools and kind of thinking about programming and so on?
That's like kind of a funny question. I've always been really interested in biology, and I was doing biology research, and one of the people told me that I might be good at bioinformatics. And so I just completely trusted them, did my undergrad in bioinformatics, absolutely loved it, and now I'm here.
That is awesome. Cool, thank you for sharing. Let's see. So there's a new question here. How are biological or technical replicates analyzed or handled in QIIME 2? Are they merged at some point or analyzed separately as separate sample IDs in the metadata? And again, a questions broadcast channel for the instructors. Is there anyone that would like to answer this one? Otherwise, I'll put someone on the spot. Bod.
Oh yeah, I knew it was coming. I was just turning off my AC so you guys could hear me. That's a great question. So the idea of dealing with replicates in microbiomes, as far as I'm concerned, it's not completely resolved. There are definitely studies that say that a single replicate is okay. It's powerful enough to detect variation in your data and so on. But from a technical perspective in QIIME 2, you can include them. However, you need to be aware when you're designing your metadata file, I think Justine said, that make sure that you keep track of a column that's specific for that subject or that participant ID, and then an additional column that will define it as a replicate. And what that will do is allow you to use certain actions in QIIME 2 to either merge them, merge the replicates, or in other instances, you manually explore that and you decide, okay, there are outliers, I'm only going to keep one of those replicates. And then that also gives you an option of excluding them and only keeping the one replicate that you would sort of use as a proxy. So from a biological perspective, a perfect solution to merging biological replicates, but they're definitely useful. And there are ways to merging them in QIIME 2. So just make sure you keep track of that in your metadata file.
Great, thanks for that, Bod. Looks like while we were answering questions here, another one of our QIIME 2 instructors joined the team, joined the call here. So I wanted to see, Zach, can I put you on in the spotlight here and introduce yourself? We can circle back too. I'll check again here in just a minute. Okay, thank you. I still need to learn how to use Skype one of these days. Chris, how about you? How did you get started in programming and command line environments, and how'd you get into this?
So I was working at Northern Arizona University as an administrator in the social work department. And I decided for fun that I was going to take a programming class. And I enjoyed it so much that at the end of the semester, I decided to quit my job and find meaningful work in something related to programming and biology. I was lucky enough to connect with the Capareso lab. I had basically never used a computer from the command line before, at least not since the late 1980s when I was playing games in DOS. And I decided when I first started with the lab that I would try out Linux just to see if I could learn something. And I have really, really enjoyed that experience. And it's been a learning curve sometimes, but most of what I do on the computer now, I do from the command line, and it's been really satisfying.
That's awesome. We're glad you joined the team too. This next question that came in is actually geared right towards you, Chris. So we have someone would like to know, what's the future look like for using QIIME 2 inside of R?
That is a really neat question. If you guys have been paying attention to the QIIME 2 forum recently, there was an announcement that we received a really awesome funding opportunity from the National Cancer Institutes. And one big part of that grant is building tooling so that people can use, essentially so that there's an R interface for QIIME 2, and so that plugin developers can write plugins in R natively, and QIIME 2 will be able to work with those plugins. So a lot of the Python R friction is going to go away, which is awesome because QIIME 2 will just be able to do QIIME 2 things with R.
I'm very excited for that future. It'll be very fun. I guess I'll have to put learning R on the to-do list, but let's see. It looks like in general we've got some questions. Maybe Greg can. Oh, he's pushed him over. Perfect.
Yeah, Slack is lagging a little bit here for me again. So I'm trying to push those over to broadcast.
Thank you for the Slack issues. We're really hoping that tomorrow will be a little bit better. So there's a question that just came in about what do I do if I've received my raw data with 2 barcode files versus 1 barcode file? And maybe I'll kind of preface with a request that we circle back to this question tomorrow after we've spent some time doing importing and demultiplexing and talking about some different raw sequencing formats and how we might work with those in QIIME 2. I think Bod has a really great lecture lined up for us about this very topic. So if you don't mind, I would suggest that maybe we revisit that one tomorrow. Let's see. I'm trying to read it and figure out where to send it. Okay, so sorry, you can't hear that. Evan was talking my ear there. Sorry. The next question that came in here is, how does QIIME 2 handle missing samples, such as one rep having 6 samples and the next only having 5 samples due to sequencing error? Greg, do you want to take that one?
I'm happy to take that one. Yeah, so QIIME 2 isn't going to do anything specific regarding your replicates, and so this is something that would really be up to you to manage. And so you would probably need to tell it in one way or another to do something special there. If you, in response to the earlier question about grouping replicates, I posted a link to the feature table group action. That action, for example, might average replicates to create a new sample that combines all of the replicates. If you were to do that, it wouldn't know that that one sample is missing. It would just average 6 samples in one case and 5 samples in the other case. If there's anything more complicated than this that you want to do with those, it'll sort of be up to you to figure out how to handle that situation. And if you have a specific question, that about your data that I'm not answering there, you can go ahead and follow up on that, and we can discuss it in your pod.
Mike, how did you get into bioinformatics and programming and all these command line tools?
I'm sorry, audio cut out for me for a sec. Were you talking to me?
I was, yeah. I was wondering, what's your story, your bioinformatics story? How'd you get into the programming side or running command line tools or, being comfortable in the terminal?
It took me a while. It started when I was doing my master's degree in phylogenetics. That's why I'm always tagged with phylogenetics and taxonomy. And I realized I did not get my start in coding by trying to solve a biological problem. It was more about being lazy and not spending 8 hours formatting tables and text files and sequence files for use. So I started my foray into this using Perl, which I don't think I'm going to go that direction again. But when I started my PhD at University of Colorado in Boulder, I was very fortunate to do a brief rotation through Rob Knight's lab when they were all in Boulder, and got to meet a bunch of great people, many of the folks on the CHIME team. And I kind of whittled away at it and learned and had to be self-taught. At the time, there wasn't many bioinformatics available, so I was kind of by necessity had to learn how to curate all this data. So that's my story.
That's great. Thank you for sharing. Let's see. Got a question here. Why is the software called CHIME? Let's see. The easy one to go with here is Greg, but I'm curious if there's anyone else who wants to jump in. Chloe, do you know what that acronym stands for? You want to share with us?
Yeah, I think I do. It's Quantitative Insights into Microbial Ecology. Isn't that what it stands for?
Yep, that's
awesome.
Correct. Yeah.
Nailed it.
Yep. And so that, the name sort of came from a roundtable discussion we had back in probably 2010 or so. And we were trying to come up with something that was pretty unique so that if people searched for it on the internet, they wouldn't, come up with 100 different software tools or other miscellaneous things. And so QIIME pretty uniquely, probably now, 10 years later, that's just about all you would find if you were to search that on Google. And, we thought it was a good representation of what we were trying to do. The quantitative insights, at the time, not much of the work was very quantitative. It was pretty qualitative. And so we really wanted to stress that. Rob Knight and Kathy Lazapone had just recently published Unifrac. And so, that was sort of taking a quantitative phylogenetic approach to relating samples. And so that word was sort of important for us to work into it. And, the rest of it just sort of grew by trying to figure out what words we wanted to fit in there and how we wanted to come up with like a unique term for searching and for naming the software.
Something with a cool logo too.
Yep. Yeah, the logo followed shortly after.
Let's see. We've got another question here, and I'm gonna put it to the whole instruction crew, so you let me know who would like to answer this one. But what is your position related to replicate samples? And so maybe this is starting to think a little bit about the study design or those sample collection procedures. Yeah, I'm going to let someone take over here.
I could take a crack at it if no one else is is dying to answer this. So I think this kind of goes back to the previous question that was asked regarding the replicates. I personally think that the issue is still unresolved, but there are some best practices, and so I'll give you my opinion, and you can take that with a grain of salt. So this isn't the perfect answer; it's just how I perceive it in the field right now. If you are working with new environments, new protocols, new sort of sample processing that you haven't done before, I think they're extremely valuable, especially because, especially with new and unknown sample types, especially if they're low biomass, for example, you're going to get a lot of issues from the sequencing and the extraction steps. And so having those replicates can really sort of reinforce what you see as either real or some technical errors. That being said, if you have a protocol that works well, that you've benchmarked it in your lab, in your facility, with the sequencing pipelines that you have, then I think it's safe to not need those technical replicates. I mean, of course, if you have them, that's great. But I know lots of high throughput labs that don't use them after having confirmed their sort of protocol and pipeline. So that's, I think, at the moment is where the field's at. That, of course, might change as more information becomes available on that. If you ask people who are working on benchmarking algorithms and so on, they will beg you to have technical replicates. But, of course, feasibility is an issue, and so that's not always an option.
Ariel, did you have anything you wanted to add to that? I couldn't tell if you were starting to chat at the beginning there or not.
No.
Okay.
It's a good answer.
We'll go with what Bod said. Looks like the next question in here is directed at Greg. What's the future of shotgun metagenomics in QIIME 2 look like?
That's a good question. We are definitely moving in that direction towards supporting shotgun metagenomics workflows in QIIME 2. So right now, there are a lot of things you could do. I sort of think of this as like the downstream analysis steps. So if you have a feature table, and we'll define that tomorrow if that's a new term for you, but like on one axis you have your samples, on the other axis you have your features. Those features might be 16S sequences, those might be functional categories in a shotgun survey, or they might just be observations of gene sequences from a shotgun survey. You can import that into QIIME 2. You can do a lot of downstream analysis with that. And so most of the visualizations that we'll end up looking at would apply to shotgun metagenomics data. But our goal really is to go upstream of that too. And so really to start with the raw sequencing data and take you through annotation or quality control, annotation of that data, and get you to that feature table, like that shotgun metagenome feature table in this case. So we've got a bunch of folks interested in this right now. It sounds like Professor Nick Bakulich, who will probably be joining us at some point during the workshop, might be very interested in leading that. And so we might be seeing some shotgun plugins coming from his lab. So this is also a big part of the goal for that project that Chris mentioned, which is funded by the National Cancer Institute. So part of that is funding our work on building shotgun metagenomics and other omics data types support in QIIME 2. So that would also include transcriptomics, metabolomics, proteomics. These are all things that we have on our radar. And like I mentioned earlier in the day, our goal here is really to make QIIME a microbiome multi-omics bioinformatics platform.
Thanks, Greg. Yeah, I'm pretty excited about what the future has to offer here for QIIME 2. It looks like that was maybe our last question there, and we are just about at the end of our daily allotted time here. So maybe at this point, it would be great to say thank you to all of our instructors, and then kind of move on to some housekeeping announcements before we break for the day. Does that sound reasonable from the instructor crew? So, like I said, let's start with a round of applause for all of the instructors today. We had a fantastic instruction crew. Working with y'all in Slack and lending a hand when you got stuck and everything. So thank you so much. We could not do that without you. I also want to send a special thank you to Evan Bolian, running our control room, taking care of all of the technical stuff. So Evan, thank you for keeping everything running smoothly for us today. Really appreciate it. And then finally, I just want to say thank you to all of you watching. We really would not be here without you, and we're really excited that that you joined us on this kind of initial adventure into running an online QIIME 2 workshop. So thank you so much for being here with us. We're really excited about everything that we have to share with you over the next couple of days. We gave you the assignment to say hello in your pod at some point. So please do that before we get started at the next session. That'll be tomorrow for us on the West Coast in North America. And we will be posting a new round of videos in the announcements channel for you shortly, probably within the next 30 minutes, maybe 30 to 60 minutes, just to be safe. And so that'll be here in our Slack channel where we've been, or in our Slack workspace where we've been working. And we will go ahead and share another playlist of videos for you to pre-watch before tomorrow. And we will also share tomorrow the YouTube link as that becomes available to us. As you saw this morning, you know, we might have a little bit of shuffling that we have to do at the last minute, but just keep an eye on Slack. So we'll start at the same time tomorrow that we started today. So whatever time that was for you in your local time zone, that's the time you should show up again for the next session. And with that, thank you very much.